Good morning, and welcome to the 9th Annual Interdisciplinary Biomedical Research Symposium. We appreciate having all of you come here on your Saturday to join in celebrating the accomplishments of our students and faculty and the research that they've been doing. To begin, I'd like to invite the president of A.T. Still University, Dr. Craig Phelps, to come forward to address our congregation here.
Thanks. Thank both of you for all you've done to make this happen and get it organized, and you're going to have a wonderful day. I was able to sit and have breakfast with some students and some physicians and today's keynote speaker, and I think it's going to be a pretty exciting day. How many have heard of TED Talks, right? Have you ever heard of that? Well, this is like the real TED Talks, right? You're really learning about innovation, what's going on. You're hearing from current scientists and current researchers and the future that you're going to hear from. And so it really is a fantastic day, and for both Truman and ATSU to participate is really a wonderful thing, and we appreciate all the work that's done. On behalf of our Board of Trustees, thank you. Innovation is always something that we're thinking about. We know to be an institution, there's three things that you do, right? You teach and you have quality education. You do service in the community, and we do great at that, and Truman does great at that. And the third is we do scholarly activity to advance the knowledge of the professions, right? So whether it's a faculty's profession or a practitioner's profession, those areas. So it is a wonderful time, and I just want to take a moment and say thank you. You'll be hearing from some wonderful folks at ATSU University, Truman, and you'll be hearing about one of our academic leaders who played a big role in this and a role in the lives of those that have been involved in this from Truman and ATSU University. You'll hear about Dr. Sargentini shortly. But once again, thank you for being here on behalf of the university.
Thank you, Dr. Phelps. I'd also like to invite the Dean of the Kirksville College of Osteopathic Medicine, Dr. Margaret Wilson, to also address us.
Thank you, Dr. Degenhart. Well, good morning and welcome. As Dr. Phelps said, we're just really excited to have you all here today. It's a great representation of our community and across campuses, and we're very, very grateful for your participation in this very rich and wonderful experience. You know, today we also will be honoring Dr. Sargentini, and I think, again, most of you in the room know that we lost Neil Sargentini in September. He passed away. He was a devoted teacher on our campus, a wonderful colleague, and just a good person. But more than anything, I think he was a passionate researcher. And you that work closely with him, maybe in his labs or up the street with his research projects, know that to be true. And that today's keynote address is now named in his honor, I think would please him very, very much. I talked to his wife, Jennifer, a couple days ago and let her know that, you know, indeed that today would be the first Neil Sargentini, PhD, keynote address at this activity. And she said she cried tears of joy because she knew that would really touch Neil, and what a great honor. So again, we gather today to learn from each other and get to share, and thank you again for being here.
So my name is Brian Degenhart. For those that don't know me, I'm the director of the A.T. Still Research Institute, which is the sponsoring organization for this program. Dr. Sargentini, as you've been hearing, he was the co-chair of this program with me, and it's hard to believe that he's not with us today. I'd like to introduce to you, though, who is going to be, who has filled in those important shoes. This is Dr. Jeremy Hauser from the Department of Anatomy here at the Kirksville College of Osteopathic Medicine. And with that, as I would do any other year, I turn it over for running of the oral speakers. So Dr. Hauser.
Thank you for coming. So today we're also going to provide each speaker, well, it's just one. We don't have one for each of you, but a yardstick, because everybody knows that Neil would use the yardstick. So it'll be up here for you, all right? If you so choose to use that, all right? But we're going to be running this as tight as we can, about 10 minutes per speech, have a couple minutes for questions. And but do realize we have all these people are presenting up in the poster session, so you'll have an opportunity to ask questions there as well. All right. So we'll try to keep this on task because we got a full day. All right. So we'll have our first speaker today, Andrea Brown. And I'm not one to read difficult words on titles. I'm an anatomist, not a biochemist, or a physiologist. I would rather not butcher what they give you. All right. So welcome, Andrea.
Oh, yeah, I don't know if I can. Okay. I'll use this in a special moment or something. Okay. All right. Good morning. Can you hear me okay? Okay. All right. My name's Andrea Brown, and I'm a second-year DO student here. And today I'll be presenting about the respiratory responses to hypoxia and the peripheral chemoreflex in Alzheimer's disease with a rat model of the disease. So I'll explain all of those words that are in the title later. So Alzheimer's disease is very well known. It's the most common cause of dementia. And most people think of the symptoms of cognitive decline and the cognitive type of symptoms, and that's actually how the diagnosis is made as well. But actually, so those symptoms come from areas of the brain like the hippocampus that pathological changes occur in, but there are also pathological changes in the brainstem, which may, in fact, occur at the same time or perhaps before the changes in those other areas of the brain, and that can cause issues with respiration, swallowing, and autonomic systems. So, for example, sleep-disordered breathing in the form of obstructive sleep apnea has been found in greater than 70% of institutionalized Alzheimer's patients. And in the late stages of Alzheimer's disease, there's also been a shortness of breath that has been observed. So this study was looking at an Alzheimer's model of rats in a respiratory control area of the brainstem in response to hypoxia. So the study used male Sprague-Deli rats and injected the lateral ventricles with a substance called streptozotocin, which is a widely used model of sporadic Alzheimer's disease. So that's the type that's more common. It's like later onset type of Alzheimer's disease. And so these rats were subjected to 2 hours of acute hypoxia of 10% oxygen. And so this is the plethysmography chamber in which that was done. Plexiglass chamber with a differential pressure transducer that was able to measure the respiratory function of the rats. And as you can see, this is kind of the timeline here. The injection of that substance and then also the control, which was a citrate buffer, was done on day -2 and 0, and then on day 21 was the hypoxia and then subsequent perfusion to fix the brains of these rats. So the area of the brainstem that we were looking at was called the nucleus tractus solitarii, and this is the first site of the peripheral chemoreflex integration to the CNS, which it also is an area of integration of other peripheral information. So this, sorry, too many things in my hands. So this shows the outlined areas. There are different coronal slices of the brainstem showing the NTS, and those measurements of millimeters are distance from bregma according to the Paxinos and Watson Rat Brain Atlas. And we analyzed these sections for CFOS, which is a marker of early cell activation in the coronal brainstem sections. So this is the data from the plethysmography. On the left there with A and B are the in a control animal before hypoxia, so at normoxia, 21% oxygen is A. You can see that the breathing went up, and the amplitude of that curve right here is the deepness of breath, or the number of breaths that were taken per minute was, you can see, is the respiratory rate. So in B, this is after 5 minutes of acute hypoxia. You could see in a normal response that the breaths become deeper and more quick And also there's a psi that you can see, which is the quick up and down breath movement there. So in C and D, the top one in black is control animals, or this is a representative sample. And then the bottom one in blue is a STZ group. And so the red lines there are to show, just help visualize the changes. So those are the same point on both of the animals. So this is the respiratory rate in response to hypoxia. So the baseline started about the same for both. And then in the control rats, the respiratory rate increased faster there at the beginning and then stayed elevated over the course of the 2 hours. And the STZ rats, it was elevated somewhat and then slowly declined over the course of 2 hours. So these are the data that show the averages of the groups in terms of respiratory rate, tidal volume, and minute ventilation, which is respiratory rate times tidal volume. So at 21% and then 10% of oxygen. So as you can see in the first graph, the respiratory rate increased in both groups, but the difference at 10% oxygen was significant in terms of STZ, which is the blue one, was lower, less of an increase in respiratory rate. And then in the tidal volume, there was a difference at the beginning. So the STZ rats had a deeper breaths at the beginning, and they both increased around the same rate. And then the minute ventilation, the slopes of those graphs are different, so it shows there's something going on there, but the actual minute ventilation wasn't significantly different. So these show the percent change from 21% to 10% oxygen. So 21%, So the first one, respiratory rate, there was a significant difference. There was a blunted increase in respiratory rate in the STZ group. There was not any significant change in tidal volume. And then the minute ventilation, there was a significant blunted increase in respiratory, in minute ventilation in the STZ group. So that says that there is a blunted peripheral chemoreflex in the STZ group. So here, I don't know how well you can see the white dots on this picture, but this is a representative slice of the NTS outlined there. The control is on the left and the STZ is on the right, and this is stained for CFOS, which is that marker of cell activation. So the neuron, or the cells that are bright white here, those are the activated cells, and this is an early marker, so this is why we had to perfuse the brains shortly after hypoxia was induced in order to determine the response quickly after hypoxia. So there's more activated cells there in the control group than in the STZ group. And then this shows the counts of the number of activated neurons in the NTS in these 4 regions of the NTS. So in the second and third slices that we looked at, which were part of the medial NTS, there was a trend for higher number of activated cells in the control group compared to the STZ group. And this is we're still working on getting more data for this. This is 4 per group. So in conclusion, the peripheral chemoreflex is blunted in STZ rats. This could be a model for some sleep apnea or other respiratory dysfunction that's observed in Alzheimer's disease. And there's also decreased cell activation in the medial NTS of the STZ rats, which could be a possible mechanism for the respiratory dysregulation. And in the future, we plan to analyze that plethysmography data over the course of the 2 hours and determine the long-term adaptation of the peripheral chemoreflex. And then we'll also be doing further immunohistochemical analysis with the CFOS staining to determine the total number of neurons, and then also increase the number of animals that we have in the group there. So I'd like to acknowledge the members of the lab, Dr. Ostrowski for supporting in this project, and also Mahima, Jack, and Chuma, who are Truman's students, some of them are here, and they helped slice the brains and do some other things with the project. And John is a biomed student in the lab. So, and then I would also like to thank Dr. Middlemas for the use of his fluorescence microscope. Any questions? Yep. Thank you.
It's an interesting work, but the question is, you are treating with streptozotocin, which also induces diabetes. So how much of is the contribution from diabetes? Because diabetes people are also more prone for depression, and they also have sleep apnea. So if you are looking for an Alzheimer model, but you are using STC, but that also induces diabetes.
Mm-hmm.
So how do you separate the one which is arising mainly from Alzheimer's disease or from diabetes?
So the way that we separate that is that this streptozotocin is injected into the brain, and it doesn't cross the blood-brain barrier. So we're using a lower dose than you would use to induce diabetes. And by doing that, we've seen this as a Alzheimer's disease model. But it's true that in Alzheimer's disease that you would see perhaps insulin resistance in the brain as well. So,
Thank you.
Liz? Oh, thought she had a hand raised. Okay. All right.
And as we said, you can always ask questions up at the poster session as well this afternoon. So our next speaker is Sarah Dobard. Get her all set. There you go.
Okay, so I'm presenting, can you hear me like this? Is this better? Like that? Yeah? Okay, so this is part of my thesis research. I'm studying the measurement of myocardial microvascular PO2 and the impact of sumatriptan. Sumatriptan is a migraine medication. It's also called Imitrex. So migraine is a neurovascular event. It's characterized by neuronal hyperexcitability and comes with symptoms such as pain, photophobia, and nausea. The World Health Organization lists migraine as the 7th most disabling disease. And then it affects a fifth of Americans and carries with it an economic burden of $13 billion, and that's from 2014 alone in the United States. So migraine occurs when the cerebral arteries become inflamed and vasodilated. This in turn impinges on the cranial nerves and sensitizes them. Triptan drugs are prescribed to treat an acute migraine attack. They are serotonergic 5-HT1B1D receptor agonists. So they cause vasoconstriction and prevent further vasodilation during migraine. However, triptan drugs are contraindicated in individuals with pre-existing cardiovascular disorders based on the premise that they cause coronary vasoconstriction, which leads me to my purpose. We wanted to determine whether sumatriptan alters the balance between coronary oxygen delivery and oxygen consumption, leading to a decrease in PO2-MV. And so PO2-MV is the partial pressure of oxygen in the microvasculature. Specifically, we're looking at the left ventricular myocardium. And PO2-MV can be related to this ratio of QO2, or oxygen delivery, over VO2 and oxygen consumption. So first of all, we wanted to determine if a bolus infusion of sumatriptan reduces myocardial PO2-MV. Second, we wanted to see if we inhibited these 5-HT1B1D receptors and if that attenuated the PO2-MV response to a bolus of sumatriptan. So surgical technique, we used female Sprague Dawley rats. I anesthetized them with sodium pentobarbital and then inserted an endotracheal tube. This allowed them to breathe spontaneously until they needed mechanical ventilation. The carotid artery, I isolated that and cannulated it, and that cannula was hooked up to a BPA, which allowed us to measure blood pressure and heart rate throughout the experiment. Next, I found the jugular vein and cannulated that, and that was how we pushed different medications through the vasculature. We maintained body temperature using a rectal thermistor. And then I made an incision in the 5th intercostal space and performed a left lateral thoracotomy. So at this point, I've induced a pneumothorax and mechanical ventilation was initiated. And using rib spreaders, I gained access to the heart. Specifically, we're looking at the left ventricle. So we placed a phosphorometer fiber optic light guide directly over the left ventricle. So the way we determine PO2 is using the phosphorescence quenching method. So we infuse this G4 phosphorescent probe into the vasculature through the jugular vein catheter. Then we placed a fiber optic probe directly over the left ventricular free wall, and it both emits and detects a signal. And then using the Stern-Vollmer relationship, we used these signals and we were able to infer PO2. So some system validation experiments. We wanted to be sure we're actually measuring left ventricular PO2-MV. So first of all, if you look at these graphs, I have time on the x-axis and then the partial pressure of oxygen on the y. And so we turned off the ventilator at 60 seconds, and you can see PO2 fell from like 65 down to maybe 21. At 90 seconds, we turned the ventilator back on, and PO2 recovered. Next, we pushed a bolus of isoproterenol, which is a nonspecific beta-adrenergic agonist. So you would expect it to increase coronary work. And so when we gave that at 60 seconds, PO2 fell and then recovered over the next several minutes. So for our specific AIM-1, does a bolus infusion of sumatriptan reduce myocardial PO2-MV? So we gave animals either sumatriptan, a milligram dissolved in 0.1 mils of saline, or we gave an animal the saline vehicle. So they both received the same volume. So this representative tracing demonstrates, if you look here, again, time on the x-axis and the partial pressure of oxygen on the y. This orange line represents saline. So when we gave saline at 60 seconds, you can see there was little to no change in the PO2. However, the blue line is sumatriptan. So when we gave that at 60 seconds, PO2 fell about 10 points and then took several minutes to recover. So this bar chart demonstrates that the baseline PO2 values between the saline group and the sumatriptan bolus group were not different. You can see that when we gave the animal saline, there was no change in the PO2. However, when we gave the animal a bolus of sumatriptan, the PO2-MV fell an average of 9 millimeters of mercury. So sumatriptan transiently decreased the PO2-MV. And also the time to 50% recovery was 288 seconds, so that's over 4 minutes. Specific AIM-2. Does inhibiting the 5-HT1B1D receptors attenuate the response to a bolus of sumatriptan? So we accomplished this using a substance called GR127935. It is a 5-HT1B1D receptor antagonist. And so this schematic shows what we did. First, we would give the animal a bolus of sumatriptan. This is called the pre-dose. Allow it to recover, and then give it either the inhibitor, the antagonist, or an equivalent volume of saline. Give it another 10 minutes to recover, and then a second bolus of sumatriptan, which we'll refer to as post. So if you look at these charts, this shows the pre and post sumatriptan dosages for the saline group, and then the GR127935 group, the antagonist. And there's no difference in baseline. But if you look over here, this is the fall in PO2 induced by sumatriptan. So after the animal received saline, PO2 fell. There was no difference in the pre and post. But on the antagonist, you can see with the first, with pre, PO2-MV fell quite a bit. However, it fell a lot less after being dosed with the antagonist. So GR127935 attenuated the sumatriptan-induced decrease in PO2-MV. So in summary, we developed a method to measure PO2-MV in the beating rat heart. We showed that a bolus infusion of sumatriptan caused a transient fall in PO2-MV. And this sumatriptan-induced fall in PO2-MV was attenuated by the 5-HT1B1D receptor antagonist GR127935. In conclusion, the sumatriptan-induced fall in myocardial PO2-MV presumably reflects coronary vasoconstriction and reduced myocardial oxygen delivery, or QO2, due to stimulation of these 5-HT1B1D receptors. So this fall in myocardial PO2MV was relatively modest and transient, and probably insufficient to markedly impair cardiac function. Any questions?
It was very nice, thank you.
Thank you.
I do have one quick question though. What would you expect to see if instead of administering it as a bolus, if you gave it as a slower drip?
The sumatriptan. Funny you should ask. That was actually part of the study. I just didn't include it here for this speech, but we did a 4-minute infusion using 2 different doses, and it was a much less pronounced fall in PO2MV. So it changed a little bit, but nothing like this bolus infusion. Welcome.
You had a 50% return to normal after 4 minutes. Did you follow your rats out to see how much before you got back to regular levels?
See that I stopped all experiments after 10 minutes, and so that's why we did the 50% recovery time, is because they didn't all recover, but we made sure that that was enough time that they would recover halfway. If that makes sense?
Thank you. Could our next speakers come up, please? I'll let you all find your presentation there. So we have 2 speakers from Truman now, Catherine Andrews and Kimberlyn Eversman.
Hello, as Dr. Hauser said, we are students from Truman State University, and we spent our summer looking at human monocytic ehrlichiosis, which is a tick-borne illness. Specifically, we were looking at the seasonality of the disease within the state of Missouri. So human monocytic ehrlichiosis is a tick-borne illness caused by the pathogen Ehrlichiosis, which works by invading our leukocytes, or white blood cells. The pathogen has multiple hosts, but the reservoir or long-term host is the white-tailed deer, which is a common species to Missouri and can also feed all 3 stages of the vector of the pathogen, which is Amblyomma americanum, or the Lone Star tick. The Lone Star tick is what passes the pathogen from host to host. So if the Lone Star tick takes a blood meal from an infected white-tailed deer and contracts the pathogen, it can pass that pathogen on to its next host in its next blood meal. So if that next host was a human, then the human has potential to contract the pathogen Ehrlichiosis and then can become infected. Humans are dead-end hosts, though, and that means that once a human has contracted the pathogen, it can't be passed on any further. If a human does become infected, the symptoms of HME are really general. You could have, it's like the common symptoms of a cold, so fever, myalgia, things like that. So it can be hard to clinically diagnose, so additional serological analyses are required to confirm a case of HME. Because the pathogen works by invading our white blood cells, it has been shown that individuals who are immunosuppressed are more likely to become diseased if they contract the pathogen. So this is a map from the Center of Disease Control and Prevention that shows the reported cases of HME in the United States from 2000 to 2013. And as you can see, cases of HME are localized in the eastern United States with darker shades of purple indicating more reported cases for that county. For a lot of infectious diseases, understanding the incidence relies on reporting methods. So in order for the CDC to get the data that they get, medical offices and public health departments have to gather this data and then submit it to the CDC. So since 1999, HME has been nationally notifiable by the CDC, meaning that states can voluntarily report disease data. So when we look at this map, you can see Iowa supposedly has no cases of HME, but that's not necessarily a reflection of the disease incidence, but more so just a reflection of different reporting methods between states. So as you can see, HME, even though it's mostly in the eastern United States, it is pretty well spread out with just a few hot spots in Missouri, Arkansas, and Oklahoma. So we were interested in looking at how the seasonality of the disease, so the start week, the peak week, and the end week, changes with different geographical locations, specifically looking at different states. But due to issues accessing the data that study would require, we decided to focus on Missouri, which, as you can see, is a pretty big hotspot for this illness.
So our overall objective was to determine and compare the seasonality of HME in two Missouri ecoregions. Those ecoregions are the Great Plains and the Eastern Temperate Forest. In the Great Plains, we have prairies and grasslands, but in the Eastern Temperate Forest, we have deciduous trees, which means trees that lose their leaves every season.
So we were able to conduct our study with a data set given to us by the Missouri Department of Health and Senior Services, and this allowed us to determine the seasonality of HME. So the data set breaks up our cases into confirmed and probable cases. A probable case is one that meets the clinical case definition, so somebody is presenting with the symptoms described earlier, but they only have supportive lab results, whereas a confirmed case is one that has confirmed lab results and meets the epidemiological status, meaning that the person's presenting with these symptoms but also has reported exposure to ticks, Lone Star ticks. So for our study, we ran it with just confirmed cases, but we also ran the same study with confirmed and probable cases and got the same result. The data presented here today is just for confirmed cases. The data set breaks our cases into the condition county, which is what we use to divide into the two ecoregions, Eastern Temperate Forest and Great Plains. Using the CDC week diagnosed, which is based on the date of diagnosis, not necessarily the date of contracting the pathogen, to calculate the seasonality, which is, again, the start, end, and peak week of the HME season. And though we were provided with 16 years of data to make our study statistically sound, we only used years with greater than or equal to 20 cases per ecoregion.
So here we have a chart of the year, the number of cases that we're seeing in all of Missouri. I'm sorry, the number of confirmed cases. Number of confirmed cases we're seeing in the forest region and then also in the plains region. You'll notice that we have an increase in the number of cases over time, and also that we continuously have more cases in the Eastern Temperate Forest region than we do in the Great Plain region. And then, as she said, we only used years where we had at least 20 or more confirmed cases in both ecoregions. So we have those highlighted here. So how we actually did this was we, for each ecoregion and for each year, we fitted a number of cases over the 52 weeks of the year. We then fitted it with a 6-degree polynomial, and that gave us the best fit and also accounts for when we see odd cases in the very beginning of the year and also the very end of the year. So then to actually find the peak week, we just simply took the maximum of this graph, and that gave us our peak week. And then the beginning and end weeks are defined as where we're seeing the greatest acceleration in the number of cases from the previous week. So to find that, we just took the second derivative of that previous graph and then the maximum before and after are defined as the beginning and end weeks of the season. So this is a chart of our overall results with our mean start week for both regions being in the middle of March, our peaks being in the middle of June, and our ends being in October. And then the duration for both of them was about 31 weeks. So as you can probably tell, we didn't find that there was a statistical difference in the two Missouri ecoregions. So because we didn't find that there was a difference, what we did was we looked at it for Missouri overall, and we did this to add a few more years to our data that we're looking at to give us a little bit stronger results. And again, they're pretty similar, but beginning in March, peak in June, and ending in October. So as I said earlier, we did see that there was an increase in the number of cases over time, and also that we were seeing more cases in the eastern temperate forest region. So we wanted to see if there was a difference in the rate at which those were increasing. So we fitted both with lines, and then we looked to see if the rate at which this one was increasing was different than this, and we did find that they were significantly different.
So in summary, we were able to determine the seasonality of HME in Missouri, which is actually really important because when a lot of people think of tick-borne illnesses, they just think of those summer months, June through August. But we found that the seasonality starts in March, peaks in June, and goes all the way through October. And in some years, there were even cases in January or December. So tick-borne illnesses are something that needs to be thought about throughout the year. And between the two regions that we studied in Missouri, the Great Plains and the Eastern Temperate Forest, there was no significant difference in seasonality, but there was an increase in case incidence for both, with a suggested difference in the rate of the disease incidence. So as far as not finding a statistical difference between Missouri ecoregions, this could be due to the fact that Missouri has a very similar climate. So it would be important to perform the same study but add a few more states and different geographical regions to it to see if there is a difference in HME seasonality based on geographic region, which was our initial goal. Another thing that we could do looking forward is to compare the activity of the vector of this disease, the Lone Star tick, to the seasonality of HME in Missouri. So HME is not transmitted transovarially, meaning that the pathogen cannot be passed on from parent to offspring. So this only leaves two life stages for the pathogen to be passed on from, and those are infected nymphs and infected adults. So there are two studies, one by Bozak and one by Collars, that looked at the activity of the Lone Star tick in northeast Missouri and southeast Missouri. And they outlined the activity here. So the activity of the nymph Lone Star tick starts towards the end of March, peaks in June, and concludes at the end of September, beginning of October. And for the adult, starts in February, peaks at the end of May, and concludes at the end of August, beginning of September. So as you can see, there is an overlap between the HME seasonality that we determined and the seasonality of these two ticks. So it might be interesting to see if there is a significant overlap between these two life stages in the disease seasonality, and if there is a significant overlap between, or a statistical difference between the overlap between the adult and the nymph, to see which life stage is more likely to pass the pathogen forward. So we would like to thank Dr. Foray and Dr. Kim at Truman State University for mentoring us on this project, the Interdisciplinary Research Community Summer Undergraduate Research Program for funding our project, and the Missouri Department of Health and Senior Services for providing us with the data set we used.
So we'll take any questions.
Yeah, it's an interesting study, and you showed that it starts in March, which coincides with the spring, and ends in October, that the fall season, but it peaks in June. So have you looked at the effect of the temperature on this? HME diseases or the tick. Because as you see, as the year increases, you see more number, and that may be contributed to the increase in temperature over the years. They say every year, last year was the hottest in history. So as the temperature increases again, it peaks in June. So have you correlated with the temperature and the incidence?
We haven't, but we modeled this study off of a paper that has been published that looked at different climatic variables and seasonality of Lyme's disease. So that would be something that we'd like to do in the future.
So just out of curiosity, so what happens to the deer which has this pathogen? What happens to the deer, whether it dies?
Oh, so deer are reservoir hosts, meaning that they can host the pathogen long term. So there are, if you look at pictures of deer with Lone Star ticks on them, you'll find hundreds of ticks, and they can carry the pathogen long term.
Thank you.
Thank you. Yep. All right. So next we are going to have oh, there we go. Get some clapping going. Need to have a little, all right, let me pass these on and I'll talk to you guys. That's a laser pointer. Other end, there we go. All right. So we have a big team up here. 4 people from Truman State Department of Biology. So Josie Emery, Lauren Galbraith. All right. Sam Leone. All right, and Catherine Stewart. Okay. So we have, let me grab that other mic, or do you already have it up there? Okay. Well, we can just have 2 and 2.
Oh, that'll work.
Hi, so we're talking about the role of NUM protein in synaptic development, synaptic plasticity, and locomotion.
Oh, goodness. I'm sorry.
So we're going to start talking about NUM. NUM is a peripheral membrane protein found in the synapse of neurons. However, its most well-known function is in stem cells during development. So when stem cells divide, they do so asymmetrically. The apical side of the cell will contain NUM and differentiate, while the side without NUM will remain stem. In cancer, NUM mutations can lead to progenitors that are differentiated but still retain some of their stemness. This is due to NUM not localizing correctly in cells. It won't localize fully to the apical side. It will localize both to the apical and basal sides. So these are the structure of the NUM protein in Drosophila melanogaster flies. That is our animal model, and also mouse. So the only difference, or the only known difference between these 2 structural domains is that Drosophila do not contain proline-rich regions, which have been found for intracellular signal transduction, but they do contain a phosphotyrosine binding domain, which will help with tyrosine kinase signaling and the NUM localization that we just previously talked about. There's also a DPF and NPF domain, which help with endocytosis and regulating interactions with proteins that mediate endocytosis.
Keep going.
Okay. So as Josie was saying, NUM is very well understood during development, but its role in the adult central nervous system is not quite understood. In fact, there's been only one publication on the subject by Zhao et al. In 2015. And just a little refresher on the cerebellum. The cerebellum is a comparator of sensory feedback input from climbing fibers, and motor output from parallel fibers. And these fibers synapse onto Purkinje cells, which then in turn form inhibitory synapses on deep cerebellar nuclei as well as vestibular nuclei. So when motion goes as planned, glutamate is released onto these Purkinje cells, and this activates calcineurin, which in turn leads to the endocytosis of AMPA glutamate receptors from the membrane. This in turn means that the next time that the Purkinje cell is stimulated, it is able to absorb less neurotransmitter, leaning towards a long-term depression, or LTD, which means that it in fact inhibits the deep cerebellar nuclei less. So in Zhao's study, he and his colleagues were looking at they were stimulating these parallel fibers and looking at the eventual current flow into the Purkinje cells in a paired pulse protocol. So they were comparing the first pulse to the second pulse. And as you can see in A, there's tracing of these two different pulses. And they were hoping to see between the two pulses a long-term depression in that the AMPA receptors were endocytosed. And as you can see in B, the gray is the control, the black is the control, and over time there is a % change from one pulse to the other, meaning that an LTD did occur, whereas in the numb knockdown, and these were mice, I forgot to mention that, there was not an LTD, meaning that without numb, endocytosis of those AMPA receptors does not occur.
Okay, so the model organism we use is Drosophila melanogaster. We use it because it has a short lifespan, so we can go through many generations in a rather quick amount of time. They also have a fully sequenced genome and well-characterized behaviors so that way we can identify abnormalities. In addition to, they have an advanced genetic toolkit, which means that they have a large amount of loss and gain of function mutations. It's easier to get double and triple mutations. And there's lots of binary expression systems that allow for transgene expression, such as the UAS-SCAL4 system.
So we take the mature third instar larva that was seen on the last slide and we dissect it. The filleted and dissected larva is seen on the left-hand side of the screen with the brain indicated at the top. The array of body wall muscles is also well defined. On the right-hand side is the neuromuscular junction we're studying. The neurons making synaptic connections with the muscle are stained with GFP. So due to the fact that complete loss of NUM is lethal, we use RNA interference combined with the UAS-SCAL4 system to selectively knock down NUM in our larva.
So due to the expression of NUM RNAi in muscle or neuron, it will cause a knockdown of the protein. So first we stain for the NUM protein to check for the efficacy of the knockdown. So the HRP is going to stain green. That's going to be the two green stains that you're seeing there, and that's going to stain neurons green. Double staining for neurons in NUM knockdown in neurons will show only background staining. That's the left red image. And then there's going to be shown an overlapping with the synapses on the right when NUM RNAi is expressed in muscle fiber. Therefore, NUM will be knocked down in muscle fiber, but you'll still see expression in the presynaptic bouton. So therefore, NUM is located presynaptically, but not postsynaptically in neurons.
All right, here we see a larval neuromuscular junction that has undergone staining for neuronal membranes. The 1B and 1S type boutons are highlighted there on the screen. And the bouton count for the RNAi line indicates a significant decrease in synapse size when NUM is knocked down. When we stimulate the nerve with an electrode and record from the muscle fiber the evoked excitatory junctional potential, which is the EJP amplitude, we also see a decrease. The frequency of spontaneously released glutamate, shown by the histogram of MEJP frequency, is decreased as well.
So like Sam was saying, Drosophila have many well-studied behaviors, and one such behavior is the locomotion of the third instar wandering larva. So for this assay, we were looking to see if there was a difference in locomotion between our control larva and our knockdown larva. So we selected these third instar larvae, which are characterized by their presence on the side of the vial. We then select either sex, as we haven't determined that it is sex-related, and we wash them and we place them on a 0.5% agarose gel and let them crawl. Larvae move by peristaltic movement, that is body wall contractions from their head to their tail. So they essentially reach out, grab the hook, or their mouth latches onto the agarose, and they pull their body forward. So intermittently, you can also see, like right now, I think that was one, the larva will pause and change directions and make a few mouth hooks and then change directions again. This is what we characterize as a search and is a sign of irregular behavior. So after we remove the larva, we can look at the mouth hook prints that they left behind, and we can analyze the path for overall path trajectory, path length, and distance between mouth hooks.
Okay, so we characterize path trajectory into 3 types. There's curved and straight, and then we have irregularity. And irregular is characterized by either more than 2 searches or a sharp angular turn of more than 90 degrees. All right, so the graph on the left is the breakdown of path trajectory. We have the control and then the numb knockdown line. Green is the irregularity, and you can see there's a significant increase in the amount of regularity sorry, in the numb line. It's 15% to 45%, I believe. And then these histograms here show path length in centimeters. Overall, there's a significant increase of the numb knockdown line versus the control. And then so I broke it down by the 3 types of trajectory, and it seems that the only one that's significant is the irregularity.
We have been able to confirm that NUM is localized presynaptically rather than postsynaptically. Our experiments with the knockdown of NUM have also shown a reduced number of synaptic boutons, a reduction in action potential dependent glutamate release, an increase in searching behavior, sharp angular turns, and locomotor activity.
So in the future, we are currently in the process of making a genetic cross where we will hopefully knock down fly wild-type NUM and replace it with wild-type mouse NUM and attempt to rescue the phenotypes we saw in our control larva. And that hopefully that will show us that indeed NUM does affect locomotion. We are also going to be trying to localize where in the bouton exactly NUM is by staining for some of the other proteins NUM is associated with.
So due to NUM's presynaptic staining, we've been investigating the active zone and periactive zone in presynaptic neurons. So neurotransmitters are going to be carried to the synapse and vesicles, which are shown in the figure below, and then Bruch Pilot is going to direct these synaptic vesicles to the membrane. Calcium is going to influx into the cell, and glutamate is going to be released. During endocytosis of glutamate will bind to the glutamate receptors dGluR2A and dGluR2B. And then clathrin-mediated endocytosis will occur, which is shown on the left side of the diagram. So in the future, we hope to find out which one of these proteins or other proteins NUM could be communicating with in neurons. So we wanted to thank Dr. Brett Burke for his guidance. We work in his lab at Truman State, and our other lab members, and of course, Truman State University's Office of Student Research.
Do we have any questions?
All right, thank you.
Thank you.
After we do two more talks, we'll take a little bit of a break. You want to help me find here? Not sure which one yours is. Which, okay. Get that up. Now do you, the top is the laser pointer. Okay. That's your forward, obviously. So you don't have to use this one. Oh, I guess I'll introduce you, right? All right, so now we have Hibba Syed from Dr. Zulfikar Ahmad's lab.
Good morning, everyone. I'd like to thank you all for joining me. My name is Hibba Syed, and I am a second-year biomedical sciences student here at KCOM. And I'd like to talk to you all today about my research, which deals with exploring the link between antimicrobial properties of venom peptides and microbial ATP synthase. So I'd like to start off by talking to you all about the major issue at hand. Antibiotic-resistant microbes have caused 700,000 deaths per year, according to the World Health Organization, and this number is estimated to increase to about 10 million deaths per year by the year 2050. And this is due to the formation of superdrugs, which have been caused because of the increased use of antibiotics. And it's a major issue because these antibiotics are starting to lose their effect, and it provides a dire need for an alternative to antibiotics in order to combat these microbial infections. My lab is looking specifically at using the ATP synthase enzyme as a molecular drug target to combat these infections. As we know, ATP synthase is an enzyme that provides cellular energy in all living organisms by producing ATP. The malfunction of ATP synthase is implicated in many diseases such as Parkinson's, Alzheimer's, cancers, and other mitochondrial myopathies. And interestingly, ATP synthase has already been used as a molecular drug target in certain FDA-approved drugs already. And some of these examples are flavanol, which is a compound found in cranberries, which has been used to selectively inhibit the ATP synthase of Streptococcus mutans in order to help with dental caries. The FDA-approved drug bedaquiline also inhibits Mycobacterium tuberculosis, and a cytotoxic agent called apoptolytin also works against cancer cells. Now, this diagram is a simplistic view of how ATP synthase works in a cell. So ATP synthase, as you can see, combines ADP and inorganic phosphate, or PI, to form ATP, which promotes cell growth. When ATP synthase is inhibited, ADP and inorganic phosphate cannot bind to form ATP, which deprives the cell of the energy required to grow and eventually leads to cell death. So for my study specifically, I'm looking at venom peptides which possess antimicrobial activity. And here are a few examples of some venom peptides that have already been researched and proven to have this antimicrobial activity. For example, wolf spider venoms called lycotoxins 1 and 2 possess antimicrobial activity against E. coli and Candida albicans ATP synthase. King cobra venom peptides called cathelicidins also selectively inhibit the E. coli ATP synthase. And more recently, antimicrobial peptides found in Komodo dragon blood also inhibit P. aeruginosa and Staph aureus ATP synthase. So now that I've kind of talked a little bit about the background, I'd like to take a step back and talk to you all about how these venom peptides bind to microbial ATP synthase. This figure right here is the ATP synthase backbone, and there are 3 binding sites that are highlighted here, although ATP synthase is known to have over 300 binding sites. In the green here is the polyphenol binding site. The blue represents the catalytic site, and the gray portion here at the bottom is what I'm interested in for my study, and is also known as the beta-del seed motif, or the peptide binding pocket. This is named beta-del seed motif because it is located on the beta subunit of the ATP synthase and consists of 7 amino acids, which is where the del seed comes from. 5 of these amino acids are negatively charged, and these negative charges are what bind to the positively charged venom peptides. This is a representation of a venom peptide that was retrieved from honeybee venom called melittin. And as you can see, it's alpha helical in nature, and the positive charges are once again what bind to the negatively charged amino acids of the beta-del seed motif, which is located on microbial ATP synthase. So now I'd like to talk a little bit about my procedure. The first step is to prepare and isolate the E. coli membrane-bound ATP synthase. And to do this, I start off by growing my E. coli until late log phase, which is when I remove the cells to harvest them by putting them in a centrifuge at around 9,500 RPM. I then lyse the cells in the French press in order to remove the intracellular contents and wash the cells in multiple buffers multiple times in ultracentrifuge at 60,000 RPM until I'm left with an isolated F1-FO membrane-bound ATP synthase. This is a very long and drawn-out process, which I've done multiple times in order to isolate multiple wild-type proteins and multiple mutants. And this is an example of a growth curve for a wild-type protein that I isolated. X-axis here is the time in hours. The y-axis is the absorbance taken at an OD 595. And as you can see, at around 6.5 hours is when the culture reached late log phase, which is when I removed the cells to harvest them. And once the protein has been isolated, I then perform inhibition assays to measure the ATP synthase activity. And this diagram here represents a simplified view of the protocol that I use to do this. So we start off by putting the protein and the venom peptide in test tubes, and we have samples, and then we also have controls, which consist of positive and negative controls. And for both the samples and the controls, we have test tubes that have 0 venom peptide, which will also be following the same protocol. So we start off by stopping the reaction of the controls, and we stop the reaction using a detergent called SDS, which denatures the ATP synthase enzyme. And then we incubate all of the test tubes in order to allow the protein and the venom peptides to bind together. Once that's happened, we then add an ATP cocktail, which is just basically ATP, and which acts as the substrate for the enzyme. We then allow the reaction to occur. And once the reaction has occurred, we then stop the reaction in the sample test tubes using the same detergent SDS. Once the reaction is stopped, we then add the reagent called Toscan-Shore reagent, or TNS reagent. This reagent binds to the liberated inorganic phosphate and turns the solution a blue color. This blue color is then measured in a spectrophotometer at OD700. The first inhibition assay we perform after we've isolated any protein is the NBD chloride. And the reason we perform it with this drug, NBD chloride, is because NBD chloride is a known and potent inhibitor of ATP synthase. And so once we isolate a protein, we see what type of inhibition is caused by the NBD chloride. And what we should be expecting is a 100% inhibition of ATP synthase, as can be seen here. The x-axis is the concentration of NBD chloride in micromolars, and the y-axis is the relative ATP synthase activity. And this acts as a sort of system validation to make sure that we've isolated the protein correctly and that our system is working in general. So this is one of the NBD chloride assays I did for a wild-type protein I isolated. Once again, x-axis is the concentration of NBD chloride in micromolars, and y-axis is relative ATP synthase activity. As you can see, I was successful in producing 100% inhibition of my wild-type protein using NBD chloride. So a secondary goal of my project is to characterize and validate the beta-del seed motif, as mentioned earlier. So as you can see, the wild-type protein has the beta-del seed motif, as mentioned earlier. And so the 2 mutants I've used, my first mutant is the PZA60, where the negatively charged amino acids have been replaced with the neutral amino acid alanine. My second mutant, PZA61, all of the amino acids have been replaced by this neutral amino acid alanine. And once again, this is to ensure that the venom peptides are actually binding to the beta-del seed motif. So now, to show some data, I'm working with about 12 venom peptides total, which consist of both naturally occurring peptides and also some modified peptides. These 12 venom peptides have a varying degree of inhibition of the ATP synthase. And I just wanted to show one naturally occurring venom peptide, which is called Latarsen-1, which is seen here. And this is taken from spider venom. And this is a little graph of my results. So the x-axis is Latarsen concentration in micromolars, and the y-axis, once again, is the relative ATP synthase activity. As you can see, this line here is my wild-type protein, which was inhibited about 40%. And this line on top is my mutant protein, PZA60, which was inhibited about 10%. And now this venom peptide is not the best inhibitor of ATP synthase, but as you can see, even with this peptide, there's still greater than 25% difference between my wild-type protein and my mutant protein. So in order to increase the potency of these venom peptides and increase the inhibition of the ATP synthase activity, we are modifying these venom peptides. And some of these modifications include the incremental addition and repositioning of positive charges, the repositioning of amino acids, increasing the alpha helicity of the venom peptides, and also the variation in the composition of the amino acids themselves. So I'd like to conclude with some overall take-home points. First of all, our studies have shown that venom peptides cause variable inhibition of ATP synthase. We've also found that the venom peptides do bind at the beta-del seed motif. The degree of inhibition of ATP synthase can be related to the position and number of positive charges on venom peptides. Modified venom peptides do enhance the degree of inhibition, And ATP synthase can be used as a molecular drug target for diseases such as cancer and microbial infections. And this is important because we know that ATP synthase is successful as a molecular drug target in order to combat the huge problem of antibiotic-resistant microbes. Thank you very much, and I'll accept any questions if there are any.
Interesting work. So you already showed in the beginning that people have shown the antimicrobial action of venoms from other species. That means they have worked out the mechanism, how it kills the bacterial system. But what you are doing is you are looking exclusively whether it has any effect on ATP. So how you can prove that it is exclusively through ATP? That means you should compare with another non-specific proteins, let's say another glycolytic enzymes, and then show whether it doesn't have effect. But you are just taking selectively and you are looking the effect of the venom peptides with the ATP synthase. That's fine. But if you want to prove that it is exclusively through ATP synthase, then you should take some other non-specific proteins and then show what happens there. Okay, but it's very interesting work.
Thank you very much. Well, we actually are specific to finding out how it binds to the ATP synthase using the beta-del seed motif. And once we've done that, we want to characterize how that inhibition what it looks like, and then we also, a step further would be to enhance that inhibition by modifying the venom peptides. So our bigger goal is kind of to enhance the venom peptides in order to inhibit the ATP synthase to a higher extent. Thank you.
Keith, Keith, if you have a question, you can. We have time.
Why does a venom have an antibacterial activity? So what is the purpose of a venom having an antibacterial activity?
Well, this is the antibacterial or antimicrobial peptides that have been found in venom are actually, they've been involved in certain animals in order to protect the animal itself. That might be attacking its prey, for example. So there's some animals that attack their prey, and in order to prevent the bacteria from the prey to be coming to the animal itself, they have evolved these antimicrobial peptides, and that's been seen in research in different papers as well.
All right, thank you. So our next speaker, and we'll take a break after this one, is Courtney Carl. She was part of the CRDP program, so the Clinical Research Development Program, and worked in Neil Sargentini's lab. So a little special guest then today, right? All right.
Thank you. Okay, thanks. All right, so as you said, my name is Courtney Carl, and I'm an undergraduate student at Truman State University. This past summer, I participated in the CRDP at the A.T. Still Research Institute, where I spent 8 weeks working with Deborah Hudman and Dr. Neil Sargentini. Today, I will be talking to you about the impact of QA gene deficiency on DNA repair-dependent processes in E. coli. First, let me give you some background to the study. In a previous study done by Dr. Sargentini's lab, radiation-sensitive gene knockouts were identified in the KO collection of E. coli in an effort to identify genes involved in DNA repair. Of the genes identified, QA was identified as being radiation-sensitive in the KO background. In order to make the results of the study more applicable to other labs, we transferred the QA deletion from the KO background to the E. coli K12 AB1157 background. Here's some more background on QA. First off, QA plays an important role in the biosynthesis pathway of cuisine. So why is cuisine and QA relevant to humans in the biomedical field? Although cuisine is not directly synthesized in humans, we do salvage cuisine from gut microflora or food in order to incorporate it into our own tRNAs. And it's also thought cuisine deficiencies are thought to be linked to tumor-grade cancers. So why did we use E. coli for the study? As a reminder, E. coli is a gram-negative rod-shaped bacteria, and it's a common model bacteria since its genes are easily manipulated, it grows quickly, so we can get our results as soon as overnight, and finally, it's safe to use.
Now that we covered the background of the study, I'll tell you how the study was conducted. First, we obtained our new E. coli strains in the AB1157 background. The wild type is SR749, and SR4360 is QA mutant that we created via transduction. The culture medium we used for the E. coli was Luria Bertani broth, abbreviated LB, and Luria Bertani broth with 1% glucose added, abbreviated LBG. We use the LBG broth because stationary phase cells, when they're grown in a glucose-rich medium, they're known to show greater survival than cells grown in the absence of glucose, and in a phenomenon known as glucose-induced resistance. Now let's go into detail about how we grew our E. coli cells. Our cells were grown in LB or LBG broth at 37 degrees Celsius with aeration either to log-thermic or stationary phase. Here is a growth curve made with our spectrophotometer set at 600 nanometers. On the x-axis, we have OD, which means optical density, or a fancy word for absorbance, at 600 nanometers. And if you aren't familiar with the terminology of bacterial growth, there's 3 phases: lag phase, exponential growth, also known as log-thermic phase, and stationary phase. Lag phase occurs when cells are adjusting to their environment and have not started growing at an exponential rate yet. This is seen at the bottom portion of the S curve. Next, we have exponential phase or logarithmic phase, where cells are growing at an exponential rate, which is seen on the more straight portion of the graph. Finally, cells go into stationary phase when the overall number of cells stops increasing since the nutrient supply is depleted and can no longer continue supporting the growth of the population. Cells were harvested at stationary phase and log-thermic phase for our study. After preparing our cell suspensions, we diluted the suspensions with phosphate buffer in order to achieve an OD calculated using data from our spectrophotometer that would give us 100 colony-forming units per 100 microliters of cells diluted by 10 to the 5th with phosphate buffer. Now we'll go over the X-ray radiation the UV radiation procedures. So our UV radiation was generated by a germicidal lamp at 254 nanometers. Also, the entire UV procedure was done under gold lighting to prevent photoreactivation, which is a light-dependent process in which E. coli can repair UV-damaged DNA. Further our procedure, first 10 milliliters of cells were suspended in phosphate buffer and placed on a rotator and rotated. Then our control sample was taken. The cell suspension was rotated in order to ensure that the cells each received an even amount of radiation. Continuing rotation, we serially irradiated the cells to 25, 50, 75, and 100 joules per meter squared. And finally, serial dilutions were done using PB prior to plating. Next, we'll go over the X-ray procedures. So our X-radiation was produced by 160 kilovolt X-ray generator. Then we used an air bubbler to aerate our cells. Well, first we put our 50 milliliter centrifuge tube containing our cell suspension in our X-ray machine and bubbled the cells for 3 minutes to take our control sample. The bubbling was again to make sure the cells were mixed up and getting an even amount of radiation. Then we continued bubbling and serially irradiated the cells at 100, 200, 300, and 400 gray, taking a sample between each irradiation. And the serial dilutions were done with PB prior to plating. Finally, we'll go over how we use methyl methane sulfonate, abbreviated as MMS. MMS is an alkylating agent that induces defense systems, DNA repair pathways, and mutagenesis in E. coli. First, we suspended our cells in 25 milliliters of phosphate buffer in an Erlenmeyer flask, out of which we took our control sample. Then MMS was added to get a 25 millimolar concentration. Then cells were put in a rotating incubator at 37 degrees Celsius at a speed of 50 rotations per minute, which is really slow. After 40 minutes, the first experimental was taken, and then after that, samples were taken every 20 minutes for a total of 160 minutes. In order to stop the action of the MMS for hurting the cells anymore, PB containing 1% sodium thiosulfate was used so the cells would not be killed by the MMS before they had a chance to repair themselves and start growing. Now for our results. Here we have a survival curve comparing the wild type and QA mutant for UV irradiation with cells grown in LB broth and harvested at stationary phase. On the x-axis, we have the UV dose in joules per meter squared, and on the y-axis, we have the surviving fraction in log. The top survival curve is the wild type, and the bottom curve is the QA mutant. There is a significant difference between the two cell types, with a p-value of 0.03. Significance was determined for all trials using a student's t-test. Next, we have a survival curve with UV radiation with cells grown in LB broth. However, these cells were harvested at log phase. And there was also a significant difference between the two types with a p-value of 0.003. Finally, we have a UV radiation curve with cells grown in LBG broth this time and harvested at stationary phase. This was not a significant difference between the two cell types with a p-value of 0.89. Now we have our X-radiation survival curve, and this was for cells grown in LB broth and harvested at stationary phase. On the x-axis, we have the X-radiation dose, and again on the y-axis, we have the surviving fraction in log, and this was not significant with a p-value of 0.74. Next, we have another X-radiation survival curve with cells grown in LB broth, but these cells were harvested at log phase. Again, these were not significantly different with a p-value of 0.06. Then we have another X-radiation survival curve, this time using LBG broth and harvesting cells at stationary phase. This was significant with a p-value of 0.02. Lastly, we have our MMS survival curve, and this was for cells grown in LB broth and harvested at log phase. And the difference between the two cell types was not significant with a p-value of 0.07. So as I had mentioned before, sensitivity was determined using a student's t-test to determine if the surviving fraction was significantly different. Overall, the sensitive treatments were UV-LB stationary phase, UV-LB log phase, and X-ray LBG stationary phase. From the data gathered, we concluded that QA plays a moderate role in cell survival, possibly controlling a backup mechanism of DNA repair or focusing on small sets of DNA damage produced by genotoxic agents. Our data showed that QA plays a greater role in UV versus X-ray survival, possibly indicating that QA has a greater impact on nucleotide excision repair rather than recombinational repair. Lastly, QA was not as sensitive in the AB1157 background as it was in the KO background. However, we suspect the change in sensitivity may be due to differences between the two parental strain backgrounds. I'd like to give a big thank you to the Clinician Researcher Development Program of the A.T. Still Research Institute for providing me with this research experience opportunity, and I'd also like to thank Dr. Sargentini and Deb for all the patience and time they put into working with me this summer. Being the last student Dr. Sargentini mentored, I'm honored to represent Dr. Sargentini's work at this conference, and I will forever be inspired by the passion Dr. Sargentini had for his work. Thank you.
Questions? We have time. Very good. All right. We'll take a, let's start at maybe 9:30 with Thomas Jackson, so we can give you guys a few minutes break to stretch your legs. All right, folks, we'll get started here. If you could take a seat, and we'll move on with our last 5 presentations. So we're a little bit ahead of schedule, so that's a good thing to have a problem of, right? So our next speaker is Thomas Jackson. He's a second-year biomedical student. So welcome him.
Means I'm special. Well, thank you, Dr. Hauser. And so the talk that I'm going to give today is about a novel application of a surgical procedure that's normally used in adults, but we're using it in juvenile rats. So just to explain the term intracerebroventricular injection, it simply means that we are injecting into the lateral ventricles of the brain. And we prefer this mode of administration because it gives us a direct form of administration directly into the brain. And so the receptors of the peptide, as I will show you later, are in the brain. And so directly administering to the brain gives it really close to the site of action. It allows us to bypass the blood-brain barrier and also avoid systemic metabolism of the peptide. This elicits an acute effect, so there's no waning of the effect of the peptide from an inefficient route of administration, such as oral or intraperitoneal. And also smaller quantities are needed. So I know for researchers, cost is a big factor in the research that you do, so it will be a lot cheaper to inject microliters versus milliliters of a peptide. And since this does require a brain surgery, there are certain specifications that we have to observe. One is that we maintain an aseptic environment, so we scrub the surgical station with 70% ethanol, and we also autoclave a tray of our surgical equipment. The rats are subjected to a constant flow of anesthesia via a veterinarian-grade isoflurane vaporizer. And there are certain drug treatments that we administer for the purpose of maintenance. So there is the anti-inflammatory glucocorticoid dexamethasone that is injected pre-surgery to prevent any swelling of the brain. The antibiotic Batril is administered post-surgery intramuscularly to prevent infection. And the analgesic buprenorphine is administered also post-surgery subcutaneously to manage pain. And finally, for the purpose of fluid reconstitution, to restore any dehydration state that may have occurred throughout the course of the surgery, and also to replace any blood loss, we give injections of isotonic saline. So just to explain the surgery that we do, so this is a stereotaxic surgery, which means that we have a movable apparatus, this thing here, that gives us measures of 3 dimensions of the surgical plane. And those 3 dimensions are anterior to posterior, medial to lateral, and depth, so from the surface of the brain down into the lateral ventricles. This micromanipulator gives us caliper measures of exact skull-brain coordinates. I will point out also that there are ear bars to hold the head steady. Those are inserted into the ear canals to prevent the head from moving throughout the course of the surgery. Also, I will point out where this clamp is. We actually have a bifurcated rostral tube. So in one of the openings, we place the rostrum of the rat, and in the other, we connect a hose from the isoflurane vaporizer to maintain anesthetization. Also, before surgery can begin, we have to shave the head from the intraoral line to the intraorbital line, and it's within this area that we make our incision. Now, I realize that intraoral to intraorbital is kind of backwards, but I'm left-handed, so that's sort of my prerogative. And in order to use stereotaxic coordinates, we must first start with bregma. Bregma is known as the intersection point of the coronal and sagittal sutures. It's also the point of junction between the frontal and parietal bones of the skull. And that's located here. For our purposes, though, no matter how you want to define it, it is the starting point for stereotaxic coordinates. So if you place these stereotaxic coordinates on a mock Cartesian coordinate system, bregma would be our 0, 0 origin. And so using a Paxinos and Watson atlas of stereotaxic coordinates for a young rat, we can measure that our lateral ventricles are 0.9 millimeters posterior from bregma, and 1.5 millimeters lateral, so in each direction. Our next step after locating the lateral ventricles is to drill burr holes into the skull so that we can expose the surface of the brain. And so the lateral ventricles will appear in approximately these locations. And once the surface of the skull is exposed, we inject at a depth of 3.6 millimeters from the surface of the brain, and that will put us directly into the bulb-like structure of the lateral ventricles. And that's our 3rd dimension of stereotaxic coordinates. And so just to talk a little bit about the application of this protocol, the project that I'm currently working on with Drs. Mittelmaus and Ostrowski asked the question, does ghrelin effectively reduce depressive-like symptoms in juvenile rats? And so for a couple of definitions, ghrelin is a brain-gut peptide that has the endocrine function of being orexigenic, which means that it stimulates the hunger sensation and drives the desire to consume food. Depression, in terms of laboratory science, can be defined as a reduced rate of neurogenesis, particularly within the hippocampus, that leads to a decrease in hippocampal volume. And so the receptor sites for ghrelin are located in the hypothalamus, the pituitary, and the hippocampus. And I highlight the hippocampus because that is the part that ties this to depression. And so these receptors in particular are the 1A isotype of the growth hormone secretagogue receptors, which are G protein coupled receptors of the Q type, so they work through the effector phospholipase C. And just for some preliminary studies that sort of stimulated our interest in this, ghrelin administration in adult rodents has led to an increased rate of neurogenesis and a decreased rate of depressive-like behavior, or a decreased incidence of depressive-like behavior. And so our question then is, does it do the same thing in juvenile studies? And so our first assessment of this question was to use a feeding behavior test. So we withdrew food 8 hours prior to surgery. And then as soon as the rats recovered from the anesthesia, we gave them a pre-measured amount of food, allowed them to eat for 2 hours, and then measured the amount of food that they consumed. And as you can see, the 500 picomolar treatment group gave us the most robust effect in terms of feeding behavior. And so that was the concentration that we decided to go with for the remainder of experiments. And so for further behavioral test, we use the force swim test. Normal behavior in the force swim test is swimming or climbing, which are kind of self-explanatory. But the depressive-like behavior is immobility time. So if you can see this first rat that is just kind of floating in the water, not really moving or doing anything, that is considered depressive-like behavior. Our last behavioral analysis is the tail suspension test. So we actually suspend the rats by their tails, and any attempts to escape, such as in pains 1 and 3, are considered normal behavior. But immobility time, such as in the middle pain, is considered depressive-like behavior. And I will point out that the tail suspension test is normally used for mice, but because juvenile rats are approximately the same size, we decided why not try it in juvenile rats. And so our results from this show that there is no statistical significance so far between the control groups and the ghrelin-treated groups. However, there is an apparent trend that actually says that there's more immobility time for the ghrelin-treated group, which, if you remember from what I said earlier, it's kind of the opposite of what we would expect to see in terms of an antidepressant. So a little disappointing, but that's science. Also with the tail suspension test, we saw the same kind of trend, but if you notice, the error bars are much smaller, which means that it could be a much more robust test and would give us a lower degree of variability. And so to conclude, ICV injections we found can successfully be used in juvenile rat studies, which was cool, that's what we were hoping for. ICV injections of acylghrelin produced an acute and immediate peptide effect. This was demonstrated by the feeding behavior test. We are currently testing ICV injections of brain-derived neurotrophic factor, or BDNF, as a potential antidepressant, and they will be administered, of course, through the ICV injection protocol. We also found that the tail suspension test has a lower variability and may actually be more effective as a behavioral test than the force swim test, which is very interesting considering that it's normally used for mice and not for rats. Future studies, we are currently looking at quantifying neurological changes to determine the effect of ghrelin on neurogenesis and mitogenesis. And someone, probably not us, but someone can look at new assessments for behavior. And with that, I would just like to acknowledge my mentors, Dr. Middlemas and Ostrowski, one of my advisors, Dr. Chang, the core imaging technician, Erica Stanley, and the laboratory technician, Ping. Thank you.
Do we have any questions? Oh, all right.
Nice job, Thomas. Quick question. Using the tail suspension, since it's new, have you done any studies with anything else to serve as a positive control to show that it can actually differentiate between those different states?
Okay, got it. No, in our lab, Erica is actually doing projects with fluoxetine, so looking at a known SSRI, so an accepted antidepressant, but as far as controls, we just use an ACSF vehicle, so that's artificial cerebrospinal fluid, and that's the vehicle for our injections. So we look at that compared to the ghrelin-treated groups.
I was thinking specifically of the tail suspension being able to differentiate a depressed versus non-depressed state.
Yes, I would say that would be what other students in the lab are looking at with the fluoxetine and control experiments.
Okay, one question. So what you described, from the way you described it appears, you really need more precision work. So congratulations for that. My question is, why did you use ghrelin, because it's a gut peptide, to study for depressive work? There are other antidepressant drugs you could have injected. Or just you want to find out because this gut peptide, they also have action, psychological action. So what is the real motive to use ghrelin to study the antidepressive action?
Well, that's a much nicer question, and I appreciate that. Our main motivation is that it is endogenously produced, so it could be more of a natural remedy for depression than, say, a selective serotonin reuptake inhibitor. Also, it has been shown in adult studies to work, so we mainly, in our lab, look at juvenile studies of depression. So we would like to potentially develop a safer method of treating depression within juvenile patients.
Thank you.
All right, our next speaker from Truman is Michaela Sadoff.
Okay, good morning, everyone. So I'm Michaela. I'm a Truman undergrad. I'm a junior. And this summer I worked with Dr. Brett Mitchell in his hypertension lab at Texas A&M's College of Medicine Department of Medical Physiology. So hypertension is officially diagnosed as having a blood pressure of 140/90. The higher the blood pressure, the harder the heart has to pump. We all know this. Hypertension is commonly referred to as the silent killer because 1 out of 6 hypertension patients are actually unaware of their own disease, mainly because there are no obvious symptoms. Hypertension greatly increases the risk for heart disease and stroke, which happen to be the first and third leading causes of death in the US today. According to the CDC, it currently affects 1 out of 3 American adults, which is a staggeringly high number. So Angiotensin II is a hormone that plays a central role in cardiovascular homeostasis. Some people with hypertension have high levels of Ang2. When given to humans and animals, Ang2 administration raises blood pressure and causes inflammation. It does this by acting on these mechanisms in this renin-angiotensin-aldosterone pathway, such as sodium retention, vasoconstriction, and immune cell infiltration. So the overview of hypertension and Ang2 naturally flows into a brief discussion of lymphatics due to its association with the immune system and inflammation. A popular analogy for the lymphatic system is comparing it to a sewer system, since it's found throughout the entire body and it's very good at getting rid of waste. Its main functions are to thus maintain homeostasis of interstitial fluid and to traffic immune cells from the interstitium to draining lymph nodes. Since lymphatic vessels are found throughout the entire body, it makes sense that the kidney would have them too. Kidneys' main functions include osmoregulation and filtration. Renal lymphatic vessels, or RLVs as I'm going to call them, are normally located only in the renal cortex surrounding arteries. The renal cortex is the layer of the kidney between the capsule and the medulla. So in kidney disease and human kidney transplants, inflammation and renal lymphangiogenesis, or the creation of new lymphatic vessels, are actually found to be linked. However, exactly how renal lymphatics are altered in Ang2-induced hypertension is currently unknown. Therefore, our question becomes, how are renal lymphatic vessels affected in the Ang2 model of hypertension? Our hypothesis is that Ang2 administration will cause hypertension, renal immune cell infiltration, kidney injury, and increased renal lymphatic vessel density. So our methods can be split into 2 main sections: inducing hypertension and analyzing tissues for RLV proliferation. We used strain C57BL6 mice, all between 10 to 12 weeks old. And to induce hypertension in our mice, we used osmotic pumps, which were implanted surgically and subcutaneously in the mouse backs. And then the osmotic pumps, and the pumps contained either angiotensin II or saline, the latter serving as our control group. So the top right corner's image, right there, that's our osmotic pump, and it has a flow moderator, which is responsible for controlled release of Ang2 into the mouse's body over the 3-week period before euthanization. So systolic blood pressure readings were taken weekly as well, using the tail cuff method, and once euthanization took place, analysis of tissues began. For this, immunofluorescence, or IF, and real-time quantitative PCR were employed. IF was used to analyze renal lymphatic vessel density, and we used the LV markers Live1 and Potoplanin. For PCR, we used that to analyze gene expression changes and markers for immune cells, kidney injury, and lymphatic vessel formation. As you can see by this graph, the weekly tail cuff blood pressure readings confirm the development of hypertension in our Ang2 mice by the 3-week mark of Ang2 administration. So this is our first PCR data graph. This is for immune cell markers. So when looking at this PCR data for gene expression of immune cell markers, we see that the gene CD4 and TBX21 were significantly expressed in our Ang2 mice. CD4 is a T cell marker, and TBX21 is a T helper 1 cell marker. So the kidney injury markers of fibronectin and ceruloplasmin were also significantly expressed in Ang2 mice, while the expression of lipocalin-2 was very close to being significant. Fibronectin is an interstitial fibrosis marker, lipocalin-2 is an early biomarker of acute kidney injury, and ceruloplasmin is a positive acute phase protein. So finally, VEGFR3 and Potoplanin were significantly expressed as well in their lymphatic vessel markers, so this suggests formation of lymphatic vessels. VEGFR3 is the VEGF-C and VEGF-D receptor. Potoplanin is a lymphatic endothelial cell marker, and VEGF-C and VEGF-D are lymphangiogenic ligands for VEGFR3. So just to remind you, lymphangiogenesis is the formation of de novo or new lymphatic vessels. So this last slide of PCR data is really exciting for our lab because while gene expression of markers for immune cells and kidney injury have already been studied in the Ang2 model of hypertension, gene expression of lymphatic vessel markers has not yet been studied in this model. So this is really new, exciting, unexplored territory for us. So now for my IF data, this is staining for lymphatic vessel markers after the 3 weeks of Ang2 administration. I want to preface real quick with an explanation of finding these. This is a very subjective process, or it can appear to be so. So when we search for RLVs on the fluorescence microscope, it's important to remember where they're going to be found, which is actually going to be around renal arteries in the renal cortex. Thus, the large lumens you see in the following pictures, that, and then I can't reach this, well, they look like that. So those are actually the renal arteries. So renal lymphatic vessels will have lumens as well, although it's possible for them to collapse or appear as bright fluorescent lines instead of hollow structures. So comparing a control mouse on the left and to an Ang2 mouse on the right, we observed that an Ang2 mouse had more renal lymphatic vessels around the artery. However, this was inconsistent amongst other areas in the same kidney slice. In some areas, renal lymphatic vessels weren't even seen and weren't increased. So this basically means we just need to go back and objectively quantify these lymphatic vessels to get better, less subjective, and more accurate comparisons between the control mice and the Ang2 mice. Because my project was preliminary this summer, this quantification had yet to take place. So again, the arteries, you'll see right there, really big, the lumen. This is Potoplanin staining, which is also a lymphatic vessel marker. And this staining confirmed what we saw in the Live1 staining around some arteries. We observed more RLVs, but that wasn't really consistent in all the areas of the tissue. So we should carefully quantify the vessels by meticulously counting them, one by one ideally. That's kind of the only way to do it right now. In conclusion, in our Ang2 hypertension model, Angiotensin II caused hypertension, renal immune cell infiltration, and kidney injury. However, renal lymphatic vessel density was not increased as much as expected and was not really consistent. This may have been due to underfixation of tissue, as histology reported a slight error in the process to us. It's also possible that 3 weeks of the Ang2 treatment was just too intense and possibly caused acute kidney injury, kidney failure, and damaging fibrosis. So an H&E staining in the future would be helpful to determine if fibrosis was the cause for a lack of lymphangiogenesis. In the future, we hope to repeat our 3-week Ang2 model with better fixation for optimal results. And we also want to apply our Ang2 model to KidVid mice, or kidney-specific VEGF-D mice, which are genetically engineered to produce new renal lymphatic vessels upon introduction of doxycycline, an antibiotic, in their drinking water to see if hypertension is prevented. So I'd like to extend a sincere thank you to my PI, Dr. Brett Mitchell, Dakshna, our grad student, Kat, our lab manager, Pauline, Mary Ann Wolf, the Texas A&M Summer Research Program, and the American Heart Association for supporting me. Thank you.
It's a good work. So it looks like you are looking for the expression of gene after administering angiotensin II, because everybody knows angiotensin II will increase the blood pressure. So actually you are looking for any new gene expression. So I'm wondering whether did you do a gene array to compare the genes that are expressed or suppressed in the lymphatic vessel compared to the one from the artery? Did you do that, or you specifically did a PCR for only selective genes?
Yeah, we specifically, we would we would have a list of genes for the day. So no, we didn't test the artery genes. It was specific to the lymphatic vessel markers and the immune cells and the kidney injury markers.
Thank you.
But that's good. We should do that.
Great talk. Were any other T helper subset markers up besides Th1?
Many were up-regulated per se or down-regulated, but significantly not so much. These were the only significant genes that were, these were significant. Okay.
Thank you. All right, today we have Catherine Jones, Dr. Catherine Jones, DO, so we'll give it to her.
Thank you. So my name's Catherine Jones. I'm a second-year general surgery resident here at Northeast Regional Medical Center. So one thing that we do once a month is tumor board. And we discuss cases that we have at the hospital. So as we've been involved in that, I've come to appreciate the role of histopathologists. They're an integral part of the multidisciplinary team. These are tissue sections of adenomatous colon polyps, and I have a few examples. These tissue samples and the diagnoses that they unwrap have a huge impact on the diagnosis and treatment of patients. Their seemingly benign tissue samples can turn into, I have a picture here. This is an adenomatous polyp here, and show that it can turn into life-altering, debilitating, life-threatening colorectal or colon cancer, sorry. So colorectal cancer has a long preclinical phase. It can take up to 10 to 15 years for a polyp to develop into colon cancer. Colonoscopies allow us to successfully detect adenomas at an early stage and detect colorectal cancer and intervene. So here's an image of what it looks like during a colonoscopy. Colorectal cancer is the third most common cancer in American men and women, and it's the second leading cause of cancer death. Colorectal cancer screening detects early stage cancer and adenomatous polyps, and it's been proven to reduce colorectal cancer mortality. So that was kind of an introduction to why our study is relevant and important. So the title is Adenoma Detection Rates at Northeast Regional Medical Center, and it's a retrospective study. My faculty supervisor was Roy R. Danks, one of my attendings, and program director of the general surgery residency program. I also was assisted by Mikkel Rogers and Mackenzie Lesh, both fourth-year medical students here. All right, so this is a slide explaining what the adenoma detection rate is. Okay, so I'm going to explain that this kind of is a diagram that shows, so say this endoscopist performed 11 colonoscopies. If he had one or more adenomatous polyps in one colonoscopy, that counts towards his or her adenoma detection rate. So in this example, even though there was 3 adenomas detected in this one colonoscopy, it only counted as 1. So here we have 1, 2, 3 out of the 11 colonoscopies that detected 1 or more adenomas. And in order to calculate that, you need to wait till the pathology results come back. So in this simple example, the ADR is 0.27 or 27%. So in the rest of my talk, I'm going to say ADR instead of adenoma detection rate. All right. So ADR is important because it's been proven to reduce the incidence and mortality of colorectal cancer. So an endoscopist that has a high ADR when compared to one with a lower ADR has been found to reduce mortality and incidence. So these are the logos for the different organizations that recommend and endorse the adenoma detection rates. So one is the American College of Gastroenterology, the American Society for Gastrointestinal Endoscopy, and the GI Quality Improvement Consortium. And it's a nationally recognized benchmark for endoscopists. Okay, so then, well, what is the benchmark? It's 25%. But interestingly, the nationally recommended ADR deemed acceptable is 25%, but there's master level ADR, which is considered 47 to 48%. So if you were the patient, if I were the patient, I would hope that I had a rock star endoscopist who had that 47% adenoma detection rate, and hopefully above the benchmark. Okay, so we studied the ADR at our institution here, and we studied it across the different specialties, because at our hospital we have family medicine endoscopists, internal medicine endoscopists, and general surgeons. The way that our method was, we took the current procedural billing codes from May 2016 to December 2016, and we included the most recent 34 colonoscopies in each of those groups. And we mined the data and read the operative reports and the pathology reports to try and make sure that we included only screening colonoscopies, and we found which colonoscopies were able to find adenomas. So let's see, what are the interesting points on this slide? Okay, so this is the ADR for family medicine, 29.4, general surgery, 26.5, and 35.3 for internal medicine. Interestingly, the male ADRs are higher than the female ADRs. The incidence of colorectal cancer is higher in males, so they're going to have more adenomatous polyps. So some of the challenges we faced was that the different endoscopists reported differently in their operative reports. So it was kind of difficult to find out which ones were actually screening colonoscopies, and that kind of leads into a future study that we're going to do that I'm going to talk about a little bit later. And another thing to point out is the ADR for the whole hospital is 30.4, which is well above the national benchmark. These are just, this is a summary of the type of polyps that were found during colonoscopies. So, sorry. This tubular adenoma with high-grade dysplasia is what we found the most of. And out of all the colonoscopies that we used in our study, there were 3 moderately differentiated adenocarcinomas. All right, so adenoma detection rate is included in 3 other quality monitors for colonoscopies. So bowel preparation is important for a colonoscopy. Cecal intubation rate. So there's been an association between a low cecal intubation rate and post-colonoscopy adenoma or colorectal cancer. So what that means is if the endoscopist made it all the way to the cecum. And then the bowel preparation quality, that kind of speaks for itself. And then adenoma detection rate, I've already explained previously. And we talked about how it decreases the incidence and mortality. All right, so the conclusions for my study, or our study, was it highlights the need for standardization of data capture for screening colonoscopies and accurate acquisition of quality data metrics. So the next study that we're going to do would require a prospective design with a standardized format for data reporting, such as a preoperative template. This is something that I designed to use in our prospective study that would make it easy to record the data, and pointing out that we would know for sure whether it was a screening colonoscopy. We would include withdrawal time. It's been proven that if the withdrawal time is too quick, you may be missing polyps on the way. And then have a standardized way of recording the bowel prep and cecal intubation rate. So whoever's doing the recording would record whether they reach the cecum or not. So the goal of our study was to emphasize the importance of the institutional ADR, raise awareness for the need for improvement in ADR, and identify barriers to quality improvement. And so colorectal cancer is, as you know, recommended for average risk patients at the age of 50 to 75, We want to make sure our practitioners at NRMC are meeting the nationally recognized benchmark and are meeting the standard of care and hold our hospital to a high quality of standard. The conclusions, ADR decreases the incidence and mortality of colorectal cancer. We want to raise the awareness for the need to improve our ADR at our hospital and identify barriers to quality improvement. The people that I would like to thank is my faculty advisor, Dr. Danks, who's here today, and Mackenzie Lesh and Mikkel Rogers, the students that helped out on this study. Thank you. So I don't know when the appropriate time for this is. I've made a handout with the current colorectal screening guidelines if you would like one. So I guess I could pass them around, and if you want one, take one.
Well, if they have, do you guys have questions for her? Looks like we have some. So I'll let you have that.
Oh, thanks.
Question, you looked at just the last few months. Did you have any numbers on your clinicians as far as how many colonoscopies they've done over a past year or over past several years to see if experience had any influence on their detection rates?
Yeah, that's a very good question, and something that came up when we were doing the study is in the time period from May to December, some of the endoscopists only performed 2 colonoscopies. So that didn't make the statistical power of our study very strong. That's why we decided to do a prospective study and include a higher number of colonoscopies and make sure each endoscopist has a high number of colonoscopies. So, yes.
You mentioned that the way they do colonoscopy is different with the different endoscopies, right? So to me, it looked more like an art than as a science, because it depends with the endoscopist. So when you said in one of the points that to improve the quality, are you planning to train the endoscopists so that you put them all on same level?
Mm-hmm.
Or because the way you said that the detection rate is different with the different endoscopies. So are you planning to improve the quality so that you give the same training or same standard for all the endoscopists?
Yeah, that's a good question. I don't know that I could control the same training, and maybe I explained it a little incorrectly. They pretty much follow the same procedure, but they report the procedure a little bit differently. There's not a standardized way of reporting the bowel prep. All of them mentioned in their reports whether they reached the cecum, so that's pretty standard. One of the big difference that I want to emphasize in the next study is the withdrawal time. So if the endoscopist takes a very long time, a short time to withdraw, they may be missing adenomas. And if they take a longer time, they'll likely see more. So maybe if in the next study we see that some endoscopists are very quick, maybe that would be something that we could address.
Thank you. Our next speaker is Cade Kinney from the biomedical program.
Hello, and thank you all for coming. Today I'll be telling you about my research titled Carbonic Anhydrase Inhibition Does Not Impact Force Generation or the Fatigue Response Following Sustained Maximal Voluntary Isometric Contractions, which is abbreviated MVIC. So fatigue for our purposes is defined as a decrease in force generation. And the graph here shows a 2-minute sustained hand grip MVIC, and we see that force begins at around 350 newtons and ends at around 70 newtons. Thus, these muscles are considered 80% fatigued. Previously, we have discovered that fatigue manifests in two different phases, with a fast phase showing a greater rate of decline compared to the slow phase, with an inflection point occurring at around 40 seconds. So during these same contractions, during these same experiments, a second set of contractions were performed after 10 minutes of rest, and here we observed two different responses. Those who recovered to produce an initial bout 2 force equal to that of bout 1, and those who did not recover. We noticed that with the recovered, which is in the blue there, it is associated with a greater rate of decline in the fast phase and an earlier onset of the slow phase. This shows that there's a neural component to the fatigue response. The failure to recover at the beginning of the initial bout 2 shows that there's an excitation-contraction coupling response, which is due to impaired calcium kinetics. The current study involves the same two-step protocol and the enzyme carbonic anhydrase. Carbonic anhydrase is responsible for the reversible hydration of carbon dioxide, so it pushes this formula here to the right. Inhibition of carbonic anhydrase is accomplished using a group of drugs called sulfonamides, which cause a diuresis, carbon dioxide retention, and a metabolic acidosis. Previous experiments in our lab and others have shown that the inhibition of carbonic anhydrase increases fatigue due to deranged calcium handling in vitro, but has been shown to have no effect on single contraction MVICs in vivo. Carbonic anhydrase inhibition has also been shown to impair sensory transmission, which could also affect the fatigue response. Therefore, our purpose is to determine carbonic anhydrase's inhibition effect on muscle activation and the fatigue response during sustained MVICs in vivo. So as I stated earlier, we used the same two-bout protocol, and healthy male subjects were treated either with 250 milligrams of acetazolamide or a placebo 14, 8, and 2 hours prior to testing in a randomized double-blind crossover design. Heart rate was measured using a lead-2 ECG, and blood pressure was measured using auscultation, which were recorded continuously throughout the contraction, or throughout the entire protocol. Force was measured using a hand dynamometer, and muscle activation was assessed using surface EMG during the contraction bouts at 1,000 hertz. EMG signals were then bandpass filtered, rectified, and integrated, which we'll show in the results. So the results. We see that with carbonic anhydrase inhibition, body weight decreased by 1.1 kilograms, which is associated with the body water loss, and that urine pH increased by 2.08 units. This is similar to previous findings. Resting heart rate and blood pressure showed no significant difference between the two treatments. And also, during the bout, before, during, and after contraction bouts, they also showed no significant difference. So for the isometric force, we see here that during both bouts, there was a similar rate of decline in both treatments, except that with the carbonic anhydrase inhibition treatment, they failed to recover to the initial bout 1 force. They also showed similar two-phase decreases, as previously mentioned, with inflection points around 40 seconds. So integrated EMG from the brachioradialis. We see similar rates of decline in both bouts during both control and the carbonic anhydrase inhibition, except that initial bout 2 levels are lower compared to initial bout 1 levels. Similar results were seen in the flexor digitorum profundus. Integrated EMG from the flexor carpi radialis showed no significant difference between the two bouts during the control or the carbonic anhydrase inhibition day. Similar results were seen in the flexor carpi ulnaris, except what was interesting is during bout 2, we actually saw an increase in the integrated EMG at the beginning, which we have not seen before. So in conclusion, carbonic anhydrase inhibition shows to have no effect on the fatigue or two-phase response during 2 bouts of sustained maximal voluntary isometric contractions. But carbonic anhydrase inhibition did appear to impair recovery of the initial bout 2 force levels. We found similar findings as previous studies in that there's a neural component which deals with feedback inhibition and an excitation contraction coupling failure which is associated with the impaired calcium kinetics. And we saw that the carbonic anhydrase inhibition exacerbates the feedback inhibition and EC coupling failure responses. So further directions for our research is to test more subjects to increase the power analysis and to include blood flow experiments to investigate its contribution to the two-phase response and feedback inhibition. Questions?
It's a good work. So now my question is, we already know once you do exercise, you get fatigue. There is oxygen debt. So on top of that, you already also mentioned in the introduction that if you inhibit carbonic anhydrase, there is more carbon dioxide retention, which is nothing but an anaerobic condition in the body. So my question is, why do you want to study exercise and see fatigue, which already going to create a condition? On top of that, why you are inducing the same fatigue condition and then try to find out what is the reason behind it? You get my point?
Okay, so we're doing this to investigate like what causes the fatigue response. So like here we're looking at if the feedback inhibition, if that affects the fatigue response or not, which we're using the carbonic anhydrase inhibition as that to impair sensory transmission.
Thank you.
If I remember correctly, acetazolamide is used to prevent, I think, altitude sickness, which isn't really related to muscle fatigue.
Yes.
But have you thought about doing hypobaric experiments with the same design?
Yes, so this is like an initial study to find out what exactly, kind of get an idea of what's going on, and then so we'll further dive into those experiments later on.
Okay, our last speaker for today is Renuka Ramachandra. And so we will be done a little early, so feel free to berate her with numerous questions. Is that how you wanted to be set up? No. She's going to talk for a half hour. No.
Thank you.
Here's your laser right there.
Okay. Thank you. Good morning, everyone. So I'll be talking today on expression of NAV 1.9 channels in Group 3 and Group 4 muscle efferents. I work with Dr. Keith Emsley in the Department of Pharmacology at KCOM. And this work was also done, part of this work was done by Tyler Marler, who was a biomed student. Right now he's a DO student, I think he's in the 3rd year. And I had also help from Christina, who was our summer student, and Ankita, who was a summer student, and right now she decided to join our lab for her biomed program. So this work was funded by NIH grant. So our lab basically focuses on exercise pressure reflex. So exercise pressure reflex is a neural mechanism that's generated in the muscle and which sends signals to the higher central command, which in turn activates other responses. So what happens when you are exercising, the muscles are contracted. So this muscle contraction decreases the oxygen content in the muscles. So the mechanical and the metabolic activators that are produced in the muscle over here send out signals or neural signals that in turn activate heart rate, increase heart rate, cardiac output, and blood pressure. So this works fine in a normal person. So what happens in a patient who has peripheral vascular disease like claudication or patients who have heart failure? So in that there is a chronic activation of this reflex pattern. So because of this chronic stimulation, there is a constant increase in heart rate, cardiac output, and blood pressure that in turn would lead to myocardial infarction in these patients. So we are interested in studying this exercise pressure reflex pattern, reflex over here. This, in short, I'll be calling it as EPR. EPR is mediated by this group 3 and group 4 afferents. These afferents have nerve terminal located in the muscles here, and they send out afferent projections, and the cell body is located in the dorsal root ganglia, specifically in lumbar 4 and 5. So we are interested in this pathway from the muscles to the dorsal root ganglia. So what are these group 3 and group 4 afferents? They are the group 3 are myelinated, small diameter exons, while the group 4 are unmyelinated, small diameter exons. The group 3 have a medium size cell body between 30 to 40 micron, while the group 4 are a smaller diameter cell body, which are less than 30 microns. Group 3 are known to be involved in pathology of heart failure, while group 4 are involved in cardiovascular pathology in patients with peripheral vascular disease. So we are interested in excitability of group 3 and group 4 afferents, and voltage-gated ion channels are responsible for excitability in this group 3 and group 4. So our lab focuses on understanding the role of voltage-gated ion channels in these afferents. One of the ion channels that we are working trying to understand is voltage-gated sodium channels, and so they're called as NAV channels up over here. And so far, we have been able to, in our lab, the previous work has shown that we have shown that there is, so we have 9 identified NAV channels, 1 to 9, and we have shown in our lab NAV 1.67 and 8 to be present in this group 3 and group 4 afferents. Currently, we are studying NAV 1.9 channels. So what's special about NAV 1.9 channels? These channels are slow activating and slow inactivating channels, and they're activated at hyperpolarized voltages as compared to the other sodium channels we know. They're not able to generate action potentials on their own, but they're able to facilitate and maintain action potential activity. So the presence of these NAV channels in group 3 and group 4 muscle afferents could basically amplify the stimulatory signals to increase exercise pressure reflex. And chronic activation of EPR enhances the cardiovascular pathologies. So as a first step, we had to identify muscle afferents. So we used fluorescent, all our experiments are done in Sprague-Dowley male rats. We used a fluorescent dye called dye-eye. We injected that into the gastrocnemius muscle and let the rat for 4 or 5 days to allow time for the dye to be transported to the dorsal root ganglia cell bodies. So this is basically we are interested in L4 and L5. Here is an example of a dye-eye labeled neuron. So for most of our experiments, for our experiments, what we use is the red labeled cells are the muscle afferents, and that's what we'll be using in further studies. So first question we asked is if the channels are expressed in these cells. To do so, we used immunohistochemistry. We used one of our main goal was to identify this channel, and the other goal was to identify if these are group 3 and group 4. So we used double immunohistochemistry technique using specific primary antibodies and different secondary antibody. So here is a representative picture. So we use peripherin, which is known to be a marker for group 4 afferents. And this is dye-eye labeled cells from the dye that we injected. So all the neurons that were co-labeled with peripherin and dye-eye group 4 afferents. So we looked for NAV 1.9 labeling in this group of cells, and we found some of them were co-labeled with NAV 1.9. So this shows that group 4 afferents do express NAV 1.9 channels, and we have controls that did not show labeling. So the next question we asked was, yes, we know it's present, are these channels functional? So for this, we used patch clamp recording technique, in which here is a cartoon version. There's a cell or a neuron of our interest. We use electrode, and we use internal and external solution that allow us to record ion channels of our interest, here NAV 1.9. I have a very typical sodium current. So if you look at this, this is called the activation phase, and this is the inactivation phase. And so we went and recorded from all the dye-eye labeled neurons that from our culture. So these were all cultured neurons that we isolated DRG and plated them onto cover slips, and we recorded from the dye-eye positive neurons. So here is an IV relationship, that is a current to voltage relationship. And if you note that some of the neurons had this little shoulder current. So if you look at the currents, when we stepped the voltage up to minus 30 or minus 50, we were able to see a slow activating and a slow inactivating current, which is typical of NAV 1.9. We had some neurons that did not show this NAV 1.9 current. So the next question we asked was, we wanted to make sure that this belonged to group 3 and group 4 afferents. So we plotted all the cells that we had recorded based upon their activation time, or the rise time here, and found that, so these are all the cells that we recorded, and this dashed line, anything about this are NAV 1.9 positive. So based on the cell size that I talked about earlier, anything below 30 is group 4. Yes, we saw a lot of cells in this range. The next question was, are there any cells in group 3? Yes, there were a few cells that were in the range of group 3, but size alone cannot be convincing, so we wanted to confirm this further using immunohistochemistry. So to confirm this, we used neurofilament 200, which is a marker for myelinated sensory neurons. And group 3 are small myelinated neurons, as I mentioned earlier, and therefore we concluded that NF200 positive neurons are, so the small NF200 positive neurons are group 3. So all the cells, this neurofilament 200, which are co-labeled with dye-eye, are group 3 muscle afferents. And this we looked for co-labeling with NAV 1.9, and we found few of the cells to be labeled, co-labeled with 1.9. All this, so we wanted to represent it. Okay, thank you. So we wanted to make sure that it was just not we wanted to quantify it. So all the cells were, the images were taken with image A, and then we counted the intensity of the labeling, and we had a threshold, and then here is an histogram representation of all the neurofilament labeled cells. So here you see that you have a neurofilament labeled cell, so the smaller neurofilament labeled cells were labeled with NAV 1.9. We also have all the di labeled cells that we call the muscle afferents, and then we have the group 3 afferents, and we see that NAV 1.9 was expressed in this group of neurons too. So in summary and conclusion, I would so we know that NAV 1.9 channel activity enhances neuronal excitability. So this enhanced neuronal excitability of the EPR exacerbates the pathology in patients with CV diseases. So we were able to show that NAV 1.0 channels are functionally expressed in group 4 afferents. An enhancement of group 4 neuronal activity has been shown to enhance myocardial infarction risk in patients with peripheral vascular disease. We were also able to show that NAV 1.0 channels are expressed in group 3 afferents, and group 3 afferents is known to enhance neuronal activity or in pathophysiology of cardiovascular diseases in heart failure patients. So, and thank you, and any questions?
Thank you, Renuka, for good presentation. So now, my question, see, in my mind, following his presentation and your presentation, so I cannot help but think that you have showed various sodium-gated channels, starting from 1.1 to 1.9. So I'm just wondering, these various channels are recruited depending upon the intensity of exercise, as one starts with the mild, moderate, and then intense and high intense exercise. So these are recruited based on that, or do you have any idea on that, or do they have a separate role, these sodium channels?
Off the top of the head, I don't know how well these channels are recruited during different phases of exercise. But what we know is these channels have different functions, like the 1.6 or 1.7 are really fast activating channels. So they would be conducting like quick responses like pain or something. While as the other NAV 1.8 or 9, they are slow activating, whereas 1.9 is slow activating. So they'll be there for sustained maintenance of the signals.
Thank
Yeah,
you.
but they do have different roles. But
Thank
I don't
you.
know how to correlate one with the exercise intensity. Thank you.
Okay, so that concludes our oral presentations. We're going to take a moment to stand and stretch, and let's plan on coming back at 11:45, so about 10-minute break, and we'll then prepare for our keynote address. Okay, if we could have everybody take a seat. If there's anybody in the hallway, if somebody can just let them know. Thank you. I'm going to give some introductory comments. Okay, so as we've said earlier, we have had a great loss with Neil Sargentini passing in September after a prolonged illness. He was an amazing fighter. He never gave up. He never gave up on his students, never gave up on his teaching. He always was a person who strove to do his best and always make the best out of circumstances. Within the Research Institute, he, as a basic scientist who is doing research in E. coli, and as a research institute where our purpose is to promote premier holistic health care and to promote the research around that and the translational research with it, he didn't initially find a niche for himself. And in 2006, we began expanding our centers, and we developed a center for mechanistic studies, and we were looking for somebody who was willing to take that lead. And he was one of those people. He was always willing to stand up and take on the project and make the best out of that. During his time is when we decided we were going to establish this symposium. We reached out with our Truman colleagues and coordinated extensively with our first year program. And then on subsequent years, he and I worked closely together. He was always the person who was making sure all the details were exactly right for every program. He also, as a center director managing personnel, we have, you know, in this world, there's always changes in personnel, and we had some changes, and it affected some of the grants, and so he rose to the occasion. And although as a microbiologist focusing on the radiation of E. coli, he was willing to take on studies similar to what we saw this morning, looking at rats and injecting things into the paws of rats, and then harvesting spinal cords and getting cells so that we could do the analysis of them. And so he once again was way often out of his comfort zone, but was willing to do that just because that was the type of person he was. We certainly want to memorialize him, not only today, but as we continue as an institute, as a symposium. And so we'd like to give you an opportunity to better know Dr. Sargentini.
I just wanted to say a few words about Dr. Sargentini as a researcher. He earned his PhD in medical microbiology from Stanford University in 1980. His dissertation topic was all the genes that control DNA repair in Escherichia coli, especially those responding to radiation damage. And in fact, he was curator of the Stanford radiobiology collection that included 4,000 different strains of Escherichia coli.
I had the distinct pleasure of working in Dr. Sargentini's lab for 8 years, and when he first hired me to work in his lab, he was just simply my boss. And then he quickly became my mentor because he had to teach me all the methodologies and how to conduct his research. And then eventually he started teaching me as well how to mentor his biomed students and other students that would come in to do research in his lab. And then about 3 years ago, he became a collaborator with me, and he allowed me to start doing my own research, which is not heard of very often, to allow someone who's supposed to be doing solely the investigators, the PI's research, to allow them to branch off on something completely unrelated. I will forever be indebted for him for allowing me to do that.
I'd like to talk to you about Neil Sargentini as an educational leader. When he first started as chair of our department was doing okay, but they weren't doing great with regard to educating our medical students. Dr. Sargentini, not only was he interested in having us become an educational leader in our college, but he was competitive. He wanted our department to do better. And because he wanted us to do better, he had a vision for improving. And his vision really resulted in us becoming a good. We went from being an okay department to being an excellent educational department for our students.
Dr. Sargentini, I truly appreciate for being a good example to me and many others. You showed me how to be a good racquetball player, a good teacher, a researcher, and above all, a good human being.
You know what I saw that I really liked about Neil was that he sought to master the things that he got involved with. Didn't always know what he was getting into initially, but he did his research, he was due diligent, he got on top of things, and then he had a well-organized and systematic approach to getting things done. That really lent itself to when he came over to play racquetball one day, and he didn't move the best for, even, I'm not sure about his athletic past, but I think it was probably mostly PhD work. So as Neil played a little racquetball, he really learned about the dimensions of the court and the angles and how to minimize his moves and cut the ball off, and kind of became better at playing this game and played with several friends a couple times a week. And so what he started to do is say, Dan, I think I can organize these 15 people playing in a league. And I'm thinking, gosh, maybe Neil, I'm not doing that well at it. And he helped us. And as Neil got more involved and held beginning clinics and was so embracing and wonderful with the students who found him to be nurturing and insightful, we had 75 and 80 students playing in the leagues by the end. For 15 or 20 years, he did this with us 3 or 4 times a year. And so we got to know Neil pretty good. I remember when Vineet Singh came to campus, and Vineet had played some squash in the past. Of course, he was in microbiology, and he went in there, and Dr. Sargentini just waylaid him, just beat him terribly. Dr. Singh also likes to master things. So Dr. Singh worked as hard as he could to get better than Dr. Sargentini, and he got in better shape.
I remember those days when you got me interested in the game of racquetball. I remember those days when you gave me 5 service chances and I still failed to score. But I had a desire to be really good in that game, and with your coaching, I soon became even better than you.
Something that was really remarkable about his career was the fact that he was able to include so many students in his research laboratory, actively doing research that was very publishable and that really benefited the students as far as their educational experience.
Dr. Sargentini was a splitter, not a lumper, and he didn't want to forget anything, and so oftentimes in his thought was it was better to include too many organisms than not enough. And so we went through and figured out where our gaps were and worked hard on making our department a better department at educating. And because of him, we are better.
And what I really admired about Dr. Sargentini was that he was always open to change, and he was always open to improvement, and he never was stagnant, and that is a rare gift. So over the course of 8 years, he was my boss, and then he was my boss and my mentor, and boss, mentor, collaborator, to eventually just one of my favorite people.
And I've learned so much from you. I am so proud to have worked with you, learned with you, and I will always be grateful for everything and all of those things. You will always be in my memory. Thank you, Dr. Sargentini.
Certainly want to thank Corey, his staff, and all that contributed to the video. And so I'd like to introduce to you the Neil J. Sargentini, PhD, Memorial Keynote Address. John Scousbow received his Bachelor's of Science degree in biochemistry from the University of Illinois. From there, he went to medical school in Chicago and received his MD degree at Rush University in 1980. He decided to go into internal medicine and went to Oregon Health Science Center, and as well as down to Christchurch in New Zealand, where he worked at Canterbury Regional Health Board and received his internal medicine degree training. From there, he went into his rheumatology fellowship and went back to Chicago at Northwestern University, and then began practice in 1987 in Minneapolis, Minnesota. In 1996, he was named director of the Center for Osteoporosis at Park Nicollet. Park Nicollet is a multi-specialty clinic there in Minneapolis who serves 500,000 patients in that area. So for those first 20 years, he was clearly focusing on what his dream was to be a clinician. But something happened, whether it was due to stirrings in his gut, burning in his soul. Maybe it was a moment of lunacy. He might have a whole variety of perspectives to this to add to it. But he decided in 2001 to begin to get more training so that not only could he practice medicine, but he could better answer questions that he felt he could not answer just solely with his clinical training. He received his master's degree and then went on to get his PhD from the University of Minnesota in the area of health services, research, and policy. He is a very active clinician and researcher in the area of osteoporosis, vertebral fractures associated with osteoporosis, particularly in men, as well as in a variety of other areas looking at cost of effectiveness and various other epidemiologic research. I am humbled by the fact that as a clinician, he has been able to publish in the last 13 years over 120 peer-reviewed journal articles, which means about 10 a year, which is just an astounding rate, and something that I think all of us would only be able to dream of. So it is certainly an honor and a privilege to introduce you to our inaugural keynote address for the Neil J. Sargentini PhD Memorial Keynote. John?
Thank you. And thank you, Brian, for that incredibly generous introduction. The honor is mine. I had not realized that I would be the first speaker now of the memorial lecture to Dr. Sargentini. So that is truly an honor. You certainly have obviously lost a dear colleague here so recently. So let's just make sure that I'm actually moving this thing right. By gum, I am. So I have no specific conflicts of interest to disclose, but I should tell you a little bit something about myself, although Brian, I guess, pretty much did it for me. I'm a clinical rheumatologist. I'm a health services researcher, and these are my major areas of where I can claim some expertise. And I put this up only because, for better or for worse, this is going to really inform a lot of what you hear. This is the experience and background from whence I come. And also, that'll include the fact that I do serve as chair of one of our 2 institutional review boards at my particular institution. So I have several particular agendas or objectives that I had laid out actually in conjunction with Brian. First of all, what the heck is a clinician scientist anyway? What does that term actually really mean? What is research anyway? I mean, we heard a lot of good basic science research this morning, and Moses, when we think of research, we think folks with their white coats on doing their experiments in the lab or doing the randomized controlled trials, et cetera, et cetera. But I would really encourage all of us to think more broadly and really think of research as really structured data inquiry to answer questions or test hypotheses. And then really get into, and this is where really the heart of my talk is intended to be, why should this matter? Why should we care? Why should we spend time, resources, energy involved in structured data inquiry? And I'll take it from both two perspectives. One is the organizational perspective, from the standpoint of healthcare delivery organization, healthcare payers, because their concerns and perspectives have driven a lot of the changes that are occurring in the landscape over the last 20, 30 years. But then also I'm going to try to make a case why it should matter to us individual clinicians. Why should we care about it? And what's in it for us? What can we gain from it? So the tension between clinical medicine and structured scientific inquiry, if I may put it that way, goes back a long, long time. So this is a journal article from some time ago where a physician, from a physician standpoint, a disease to be cured or prevented. This was, by the way, published in 1900 in Nature. And this is individual patients one by one, whereas investigator establishes causal relations. This is just directly quoted from the article, this particular author's perspective, between a disease and conditions that give rise to them, which really implies the study of health and human pathology at the population level. Now, when we come to a second publication, 101 years later, now up in 2001, it doesn't really look a whole lot different. Where aim of medicine, in the words of this author, is promoting health through prevention and treatment of disease, one individual patient at a time, using pattern recognition based on experience, and where the long tradition of medical training has been really based on the apprentice model, where you learn one-on-one with your mentors and so on. Whereas the aim of science, now somewhat reformulated here as the methodological and systematic acquisition of knowledge. So what I would propose is marrying these two in the following way. A clinician is a health service professional, be it an MD, a DO, a physician's assistant, a dentist, anybody who is really involved in interacting with patients for the benefit of their health and welfare, delivering care to individual patients. But then the scientist piece of it is having a working knowledge of scientific methods sufficient to be able to evaluate clinical research in a critical way and to evaluate quality improvement studies. And ideally, at least some of them, being actively engaged in projects to answer questions using data analysis. And so to try to piece this together, what I'm trying to show you here is kind of over the whole broad spectrum of how research can play out. We heard a lot of nice basic science this morning, and certainly this has been where a lot of research, have I got a pointer here? Top button, thank you. Okay, excellent. Basic science of discovery. And that leads to the translation chasm 1 in terms of where then that leads to proposed human applications, be it of a diagnostic test, be it of a treatment, and so on. And then typically we have what is usually in the form of randomized controlled trials to try to show that in fact that proposed human application has a proven clinical application to somebody, somewhere, some group of patients, somewhere. But then we have a lot of other ways to go to know ultimately what is the actual impact that we're having on patients that we see and for whose care we are charged. Translation chasm 3 is how do you then take a proven clinical application and actually put it into clinical practice? And translation chasm 4 is what are the outcomes patients experiences, what is the impact on their health and healthcare costs. And so this is where my background will lead to the bias you're going to hear from this talk. I'm not a basic science researcher. I'm a health services researcher. So I operate down in this sphere here. That said, clinicians can and should be important and involved and interested in what's going back up here, because ultimately, most of us want, I dare say all of us, want our work to be relevant. I mean, none of us are interested in publishing our results in the journal of irrelevant material, right? So we always want our stuff eventually to have impact. So clinicians have a role to play here. However, down here, it's essential. This does not happen without clinicians being involved in this. They're absolutely essential and central to that effort. So, why is all this necessary? Why should we bother all of this? And I'm going to start with the organizational perspective from healthcare payers and healthcare delivery organizations. And the concerns and the need for it has particularly risen more acutely in the context of rising healthcare costs. And we've all seen variations of this graph, which shows that healthcare costs per capita are rising across the developed world, particularly in our own country. But at the same time, through a variety of, through a long research agenda and cumulative evidence, that we do have significant gaps in quality of care and in outcomes of care. And I'll show you a couple examples of those. That led the Institute of Medicine to publish a very long treatise back in 1998 called To Err is Human. And this was getting into issues of errors in surgery, substantial variations in quality of clinical practice. And then a subsequent publication that you see here, Crossing the Quality Chasm. And they had a variety of recommendations that they had that we needed as a nation, as a national health agenda to really take on, to really help us with some of this. And recommendation 4.5 is we need evidence-based decision-making. Patients should receive care based on the best available scientific knowledge. And care should not vary illogically from clinician to clinician, from place to place. Doesn't mean it shouldn't vary, but it should vary for logical reasons that make sense when it does vary. So, what is some of the evidence that really gets at this particular issue? Well, there's wide geographic variation in healthcare spending in the US. And there's no clear evidence that that variation, that higher spending leads to higher quality care, better outcomes. And I'm going to show you just one example of a large body of evidence that really goes along with that. And then it really gets at the issue then, are we getting good value for our healthcare dollars? So this is this particular study really looked at healthcare spending following a myocardial infarction. And this is in the Medicare population, but it's standardized costs, meaning that really this takes out the fact that healthcare inevitably is going to be more expensive in New York City than it is in Oklahoma City because, of course, labor and capital price inputs are far higher in certain areas of the country than they are otherwise. This takes that out, and this is per person over the year following a myocardial infarction, the huge variation you see in healthcare costs across the country. And now, does this lead to better outcomes and better healthcare practices, the spending? Now, I chose myocardial infarction because it's one of those conditions where for acute MI, there is at least, to compare to some other illnesses, much broader, much tighter consensus and higher levels of evidence of what good practices are and should be done. So let's look at a couple of those. So how about the prescription of beta blocker at discharge? Well, as you can see here, it didn't matter whether you were in a low spending region or high spending region in terms of what proportion of individuals were prescribed a beta blocker at discharge. It didn't really matter whether it was a teaching hospital or a community hospital. Similarly, if someone came in with an acute coronary event, was there an attempt made to get them reperfused, to open up that artery within 12 hours? Well, it didn't really matter whether you're in a low spending region or a high spending region. So this is just one example of a very wide body of evidence to suggest that there were substantial issues in terms of healthcare delivery and outcomes. So now that's for myocardial infarction, where you've got a very deep body of literature in terms of good practices and that kind of things. Go venture into so many other areas of medicine, and the weeds get even a lot thicker. Because here, a lot of guideline recommendations, when you look at them, they're based on expert opinion. They have to be, because we simply don't have good, high-quality available evidence in really informing us what the right things to do might really be. Standardized, blinded, randomized controlled trials, they certainly are the highest quality of evidence in some respects, but they have limitations. They have significant limitations. They are showing intervention efficacy under controlled, often ideal conditions. Again, they're showing a clinical application. They really fit into that translation chasm 2 I showed. And they're often done for regulatory approval, really, not to specifically improve care for one specific in practice. So what are the knowledge gaps left, once you're done with these things? Well, study findings often are not generalizable to the broader clinical populations in usual clinical practice settings. I'm just reminded that when I think in the osteoporosis field and I look at the inclusion and exclusion criteria for some of the main medications we use to prevent fractures, it's only a minority of my patients who would have qualified for entry into those trials. So, you got a really major issue in terms of how you apply these things. They generally show performance of diagnostic tests of intervention compared to doing nothing. But when you've got alternatives, what is the best practice amongst available alternatives? And so when you get to translation chasm 2, there is usually a dearth of evidence most of the time to really tell you that. And then what patient characteristics predicts outcomes? Is intervention A better for some patients and intervention B better for others? So, and then finally, once you even have worked out some of those things, what's the most efficient way in clinical practice settings of case identification, identifying who should get intervention A or intervention B? What's the most efficient way of delivering the intended and indicated care to these patients? And finally, how are you actually doing? How are you actually performing? And then that should also need to include outcomes relevant from the patient and other stakeholder perspectives. And yes, I'm going to bring it up, I'm sorry, but it does need to include consideration of costs. I'm sorry, sometimes it's the elephant in the room. And I think that we're foolish to say that that just does not matter. So, for translation chasm 3 and 4 here, there are other types of data inquiry that have really gained a lot of traction and resonance over the last 20 years. And I'll go through each of these. They overlap. One is comparative effectiveness research here. And then there's quality improvement and implementation and adoption research. And practice-based research is sort of an amalgam of these. And I'll define each of these and illustrate the principles of them. And then as I go through those, I'm going to show you examples of 4 studies. 2 of which are my own that I've been involved in from a quality improvement and practice performance perspective. And I part chose those to illustrate as well that these can come up from questions from clinicians in the context of their own clinical practice. And that sometimes you don't need a huge $2 million NIH grant to address them, okay? That there really is room, if you will, for clinicians, I think, to step up in this regard. So, what are the objectives of comparative effectiveness research? And if anyone has the time and inclination, this is a great document to really go through that I think from the Institute of Medicine that really, outlines a lot of that. And it really comparative effectiveness research falls into 2 kind of broad buckets. One is informing clinical decisions from the patient perspective, healthcare of individual patients, screening, prevention, diagnosis, and therapy. But then also another broad bucket informing health policy decisions from the population perspective. And that also includes developing the methodology of knowledge synthesis and transfer strategies, hence the whole development and refinement of meta-analysis and systematic reviews that the Agency for Healthcare Research and Quality employs so much and funds so much over the last 20 years. And then again, the organization, delivery, and payment for healthcare services, and what effect that has on subsequent outcomes. When you get to comparative effectiveness in research, practice-based research, quality improvement, now you got to really think carefully about stakeholders. Who really has a stake in what the outcomes are here and what you do with them? So a huge mistake of researchers sometimes when they want to develop a new clinical practice protocol or this kind of thing, and I've seen healthcare delivery organization administrators do this more than once, where they don't involve the people in the trenches, the clinicians delivering the care, who you actually expect to carry it out. So you really have to think about stakeholders and how the various things are going to play out from their perspective. And I'll show you a couple of examples in our work where we've done that. The stakeholders are important from the get-go. It even gets at what is the priority setting. I mean, we don't have the funds, we don't have the societal resources to research everything. We have to pick and choose. I mean, you do have to have an agenda in terms of what are the highest priority sorts of issues and topics that really are deserving of research dollars and resources. Clinicians need to be involved in that. I mean, they clearly, that's really clearly important. It gets at the issues of study design. No study, no clinical research study is perfect. Everyone has flaws. And there's always trade-offs that have to be made when you're designing a study in terms of generalizability versus rigor. And what the implications that are in terms of what you might find and how sound the results may be is really important. And then in terms of implementation of the results. So the whole idea, again, here is that you're going to be producing a product that, in fact, is implementable in clinical practice or by healthcare delivery organization. Comparative effective research, at its heart, typically is comparing the benefits and harms of two or more interventions, each with the potential to be a best practice. The comparative strategy may be nothing more than usual care or watchful waiting. If you have an elevated PSA, should you intervene right away? Should you do biopsy of prostate right away? Or sitting back and watching might be appropriate. That's just an example of a comparative effectiveness practice question. And that typically is occurring in usual ordinary practice settings. As in standard randomized controlled trials, yes, you're going to see population-level average effects in different intervention strategy groups. But they also really want to try to get the consideration of specific subgroups. What patient characteristics influence effectiveness and harms? What works best for whom? Can we help individualize decisions? Well-done CER studies can help with that to some degree. Now, there's 6 billion of us on the planet. I mean, there's just too many different subsets of variations of human characteristics to get at all of this. But at least if we think like this, we can start to think in terms of what are the most important subgroups for whom we need to ask these questions separately. Comparative effectiveness research really typically then is going to emphasize outcomes that really repeatedly now over several decades have shown to be important to patients. Pain, functional limitations, social roles. Sometimes avoidance of medication or surgery is an outcome important to some patients, and costs. Comparative effectiveness research employs any and all study designs. It can be a randomized trial. It can be a practical randomized controlled trial, and I'll show you an example of that. The units of randomization may be individual patients. It may be clinics, where different clinics are randomly assigned to certain different types of treatment strategies. It can be observational designs, and I'll show you an example of one of those as well. So this is the first example I'll show you, which is, I think, just one of the, a very high-profile study, the SPRINT trial, that was published in the New England Journal a couple of years ago that I think illustrates a lot of the principle of comparative effectiveness research. So the research question was, does intense medication treatment, where the target systolic blood pressure is 120 rather than 140, does that impact favorably or unfavorably cardiovascular disease outcomes and mortality? And so this was a randomized trial of nearly 10,000 individuals randomly assigned to intensive treatment or usual care treatment. Now, the intervention strategies are not a medicine and a matching placebo. They're treatment strategies. And so this was the intense treatment strategy, where they come in, they're started on a particular medicine. The doctor, seeing this study patient, has some latitude in terms of which drugs are chosen. They have some constraints on them, but they have some latitude in terms of what's chosen. The issue here is not so much specifically what medication you're using, is are you achieving the target blood pressure goal? And whereas in the control group, the target was left at 140. Unit randomization was the individual patient. Again, this is not a full, completely double-blind, randomized controlled trial. The outcome adjudicators were blinded to treatment assignment, as they needed to be, because you think that a cardiovascular outcome is really easy to determine. It's absolutely crystal clear who has it, who doesn't. No, that's not quite true. There is some subjectivity sometimes in adjudicating whether or not a cardiovascular outcome has actually occurred or not, or if the person died, was it actually due to cardiovascular disease or wasn't it? So they needed to be blind, but patients were not blind to treatment assignment. Providers were not blind to treatment assignment here. And then an important check on the quality of the study and the adequacy of the study design is what were the actual blood pressure outcomes in SPRINT. And as you can see here, basically they were pretty good. The intensive treatment group actually on average didn't get quite to 120. I think the mean systolic blood pressure in the treatment group actually is about 121 or 122. They got close, but they didn't quite get there. They did do a subgroup analysis. This was all pre-planned. So they wanted to ask, does it matter whether you have chronic disease of the kidney or no? Does age matter? Do we see differences in outcomes in those over age 75 or younger than age 75? Are there differences in terms of outcomes in terms of sex or race, previous cardiovascular disease, starting systolic blood pressure? My point isn't to argue what the specific data, just to really show you the principle of what they're trying to do here, is to really try to really make this as clinically applicable to actual patients that you might be seeing in clinical practice. They looked at a variety of patient-reported outcomes. I'm just showing you two of them here, medication adherence, and also then satisfaction with blood pressure care. It's basically a survey instrument of patients. Are you pretty happy with the way you were treated by your doc in terms of your blood pressure care? No, they didn't see any differences between them. My point being is that this was an important outcome of the study. Subsequent investigations really tried to then ask the question, how generalizable is SPRINT? I mean, of the whole swath of the population out there with hypertension, to what proportion do these apply? Now, they knew there was not going to be a majority because they specifically excluded individuals with diabetes. There is concern that there was more concern about the safety, I think, in terms of driving blood pressure down this low. And they were particularly concerned about safety from that standpoint in individuals with a prior history of stroke or congestive heart failure. But even still, in this particular international registry, they established that this was that basically about 34% of all these individuals would have qualified for entry into the SPRINT trial. So not ideal. This doesn't answer every question out there by any means. But at least they really looked at this. It was an important part of this to really look at this. So now I'm going to show you an example from the other major bucket of comparative effectiveness research, and that is at the population level health policy. And in this particular study, what the researchers asked was, how did Medicaid expansion in 3 states impact mortality compared to neighboring control states? And I should right away state, this is not Medicaid expansion at the time the Affordable Care Act passed in 2009. This is an earlier expansion in 3 states, Arizona, New York, and Maine. And all 3 of those states did expand their Medicaid criteria eligibility somewhat in the years 2000 to 2003, I think. And so the unit of analysis was counted, the county level mortality in those 3 states and 3 adjacent states that did not change their Medicaid eligibility criteria. And they used a time series approach. This is not a randomized controlled trial. This is a natural quasi-experiment within control county level covariates of age group, sex rates, poverty rate, unemployment rate, and median income. So this slide really illustrates what the analytic design is. This is what we call a differences and differences approach, where you really have 2 sets of controls here. You have a control group, and you have the expansion states. But within each of those, you're also comparing post-implementation mortality rates and pre-implementation mortality rates. And so, short of true randomized control design, this is one of the more powerful quasi-experimental studies that you can do. And in this particular instance, what they did show was that there was evidence of a multivariable adjusted change in expansion states versus non-expansion states, about a drop of 6 to 7% all-cause mortality. So let me move on to those other 2 buckets of research for translation chasms 3 and 4 that I mentioned, quality improvement and practice-based research. So these are questions that come up a lot. What's a quality improvement project and what's a research project? So when we think of quality improvement, historically we're really thought of as a systematic dedicated activity, but really designed within a particular healthcare organization for their internal use. How can we do our job better? How can we achieve our outcomes better, within our own particular institution? Whereas research is a systematic investigation designed to develop or contribute to generalizable knowledge. And the knowledge-seeking enterprise, by and large, is independent of normal healthcare delivery. Practice-based research is really research done in the context of usual medical care. This is sounding a lot like comparative effectiveness research, project with planned input from all stakeholders, but a commitment to actually implement the findings of the study. So something like the SPRINT study wasn't a practice-based research study. I mean, there wasn't they didn't have all the participants in SPRINT weren't there saying, okay, based on this, we're committed that we're going to implement this particular new care policy. Practice-based research really marries it with quality improvement. We're simply using the research component to really tell you what should our policy within our healthcare organization be in caring for X or caring for Y group of patients. Now, this means the clinicians own the question. So again, I'm pointing a finger at myself on this to some degree. Researchers, sometimes we can get involved in these studies, asked to bring on our expertise, and then we hear the thing and we go, boy, that's an interesting question, but boy, this question would be a lot more interesting if we phrased it this way, if we looked at it instead in this way. No. The clinician has to own this question. And we can help refine them in that kind of thing, but it always has to come back. Is the question still really getting at the issue in practice that had led to your concern in the first place? So these are all different sides of the same coin: comparative effectiveness research, practice-based research, and quality improvement. They all seek ultimately to discover ways to improve outcomes, improve our patients' experience, lower costs. The main audience for quality improvement is internal. The main audience for research is external researchers. But all our efforts, I'm sorry that that's sitting there in the way, it says all efforts seek accurate questions that are not confounding or misleading. I mean, I have some people, quality improvement practitioners, will come to us and say, well, gee, I don't want to do research. I just want to do, this quality improvement thing. And don't talk to me about data analysis or what strategies I got to have. And I say, well, are you happy with a misleading or confounded answer? Are you happy with that? And generally they go, no, I want an accurate answer. Well, okay, then we have to think about what we need to do to actually get you an accurate answer. So ultimately, if I'm asked, is my project a quality improvement project or a research project, how do I decide which it is? And I really come back to them and say, I think they're more important questions. First, what are you actually asking? What is the question to which you actually want an answer? What's required to get a credible, accurate answer to the question? What data do you need? What's the appropriate data collection strategy? What's the appropriate data analysis strategy to get a reasonably accurate question? What's the cost of getting a wrong answer, implementing a policy based on that wrong answer? Now, in the spirit of a very broad definition of structured data inquiry, I'll give you a very simple example from my own home institution. So, Park Nickel Clinic put a new phone system, to answer the phones back in, I think it was 1998 or something. It was a long time ago. And the administrators got all excited about it, and they were hearing all sorts of fad words of response times and this kind of thing. And I think the clinic spent $2.5 million in this thing. Totally failed. What data did they not collect? They didn't talk to the receptionists who actually answer the phones and might have been able to tell them something about this. So here's a very simple thing. You wouldn't think of this in the context of research at all, but they should have done some structured data inquiry in terms of really trying to get an accurate, credible answer to what they really wanted to do. So there is a cost to getting a wrong answer, even if it's a simple clinical process of, gee, well, we're going to have our staff do X and Y and Z additional things to do it. That comes with a cost. And so I think you always have to ask, well, if I get it wrong, how much trouble does that lead to? And then in terms from the IRB perspective, what are the risks to those who are being studied? So, this is where I'm going to, I'll get to that in a moment. But then how rigorous do projects need to be? Well, what are the threats to the validity of the answers that you may get? There are confounders that may, and these are variables that link to both exposure and outcomes that may bias the estimate of the association between exposures and outcomes. Secular trends. There may be things going on in the background that influence what outcomes that they're looking at. And the big kahuna is selection bias. And outside of a randomized controlled setting, this is the big one that always has to be dealt with and has to be really thought through very, very carefully. And yes, even in a quality improvement project, sorry guys, sometimes people with some statistical knowledge need to be involved in designing the project so that you're more likely to get a reasonably accurate answer. So yes, we use regression methods to control for confounders, and we have a variety of ways that are used to varying degrees of success to control for selection bias. None of these are perfect by any means. All of them have their weaknesses and limits to them. And then for accounting for secular trends, time series analyses can be a powerful analytic strategy. So the quality improvement practice-based research toolkit usually does involve the electronic health record. Surveys often can be done. Claims data, if you want to look at healthcare utilization costs, can be very important. And sometimes, yes, important questions are best answered with semi-structured interviews and focus groups. There's a whole area of research called qualitative research. Not all research is quantitative. And one of the things you lose with quantitative approaches is that you lose some of the richness of people's experiences sometimes. And so that sometimes can be an important thing to do. So when you do studies that involve the electronic health record, one thing you got to remember is that data was not collected with research in mind. It was collected to care for patients. So you have to ask yourself carefully, what do these variables mean that I'm proving? I'll give you an example from my own research. This was for my PhD, where I was doing a mixed electronic health record and survey study of medication adherence to osteoporosis medications. And one of the things I wanted to know was their racial background, what their ethnic group was. And I could have gotten that right from the EHR. I chose instead to do it by survey. Why? Because I learned at that time the way we were recording race at my clinic was that the person would come to the front desk to check in for their appointment, and the receptionist would guess. Lousy variable, terrible variable. So you have to really understand, when you're using data that was collected for other purposes from which you're using it, you really have to understand what those variables are and how they were collected. Surveys can be very helpful. You do need survey data expertise. I think it's a serious mistake for anyone in doing these kinds of things to say, well, I just have an idea of what questions I want to ask. There actually is a detailed science and depth to good survey research in terms of how you formulate, how you ask questions. And if you're really going to put a scale together, you have to have someone skilled or develop the skill sets yourself to really figure out how to establish a scale with good psychometric properties and to know what those properties are. And of course, what mode should you use? Mail, telephone, internet, advantages and disadvantages to all of those. Claims data, they can be very helpful for studies that evaluate comprehensive utilization, total cost of care. And like I say, I already mentioned semi-structured interviews here that really can be very, very, very helpful. When is IRB review needed? So now I'm putting my IRB hat on. Okay. So you need it if patients are being exposed to more risk than they would if they're getting routine care. If it's truly no greater risk than when they just step in the door of your clinic for an appointment, then you may not need IRB review. There are certain areas where you clearly have crossed a line and you need IRB to be involved. If you have patient-level interventions that are not part of standard care, if you've got truly an experimental approach, that needs IRB. If you are selecting intervention in a way that's not done in routine care, like random assignment, you got to do it. That doesn't mean you can't have control groups where you're not doing the intervention, others where you are doing the intervention. It just has to make sense and be consistent with the way you might do it in the real world. So, for example, if you figure out, well, gee, we have a new way if we're going to have our nurses call individuals a week after their appointment and to make sure they're following up, et cetera, and see what effect that has on adherence and outcomes. Well, you can say, gee, there's a real cost to this intervention. We're going to beta test it, intervene it only in clinics A, B, and C to start with, and then if it's looking good, then we'll go ahead and spread it out from clinics D through Z. That is fine. That doesn't necessarily mean that you've really crossed into an experimental realm. And a collection of data of personal identifiers that is not part of routine care. This comes up with surveys. You're not crossed a line if you're using a survey in terms of what's outside of the firewall, what's protected, like the electronic health record, if it has no personal identifiers on it. Now that means even date of birth. Can't have date of birth on there. You can have year of birth, not date of birth. And then as long as the crosswalk between the electronic health record and the survey is as well protected as the electronic health record data itself is, then at least you can make the case to the IRB, I'm not exposing folk to any more risk than if they just walked in the door for an appointment. Intent to publish does not mean IRB approval is required. If you come up with a quality improvement thing, it's intended for internal audiences, and it's a really great thing, and you got great results. Are you going to stay quiet? I think you're going to want to tell some folk, right? So sometimes that means giving presentations in meetings outside your institution, might even mean submitting a journal article to as well. So the one thing that you do need to be aware of is that many journals will require that the IRB at least had a look at the project and either approved it or declared it to be exempt from IRB review. So I'm going to show you some examples of quality improvement projects. And these are two of my own examples. And my intent here is not to convince you of the hypothesis we tested or any particular conclusions we drew in these studies, simply to illustrate some of these principles. So one of the things that I am involved in is vertebral fracture detection in clinical practice. And we can do this directly on bone density machines. We can get near X-ray quality images of the spine with them lying on their side and the bone densitometer. And the advantage is that we can do it right at the time of a bone density test. It takes my technologist 120 seconds to roll them on their side and get this additional image. So there's really a lot to like in terms of efficiency. Why is it important? Because at any given level of bone mineral density, if you have a prevalent radiographic or prevalent vertebral fracture, your risk of subsequent fractures rises considerably. It tells us a lot about bone quality independent of bone mineral density and other markers of bone quality and strength. And thirdly, only a third of these to a quarter of them are recognized clinically at the time that they happen. It doesn't mean that 75% of them are asymptomatic. Many of people who get these will get some back pain. They write it off to I pulled a muscle, I did something funny, and they just let it ride. They never get really attention for it. But so for the 70% or so of these that are not recognized clinically at the time that they happen, you have to spine imaging to actually see them. And so this is an example of a lateral spine image on bone density machines that shows you identification of a grade 2 vertebral fracture in the lumbar region here. So what we decided was to put a standing order in our institution to look for these. We didn't want to do it in everybody, because if you have a pretest probability of only 1% that you've got a prevalent vertebral fracture, I'm not sure I'm at good grounds ethically to be exposing you to even minimal radiation to really look for something where the odds I'm going to find it are so tiny. But based on a variety of research studies that we had done, we had a pretty good idea that if you met these criteria, that you had a pretest probability that's going to find something of 10% or more. So I had a good chance that if for individuals coming in for bone density test, if they have these, that we're going to find it. But then I talked to my colleagues in 2 other institutions who were very much involved in the science and epidemiology of vertebral fractures, and they weren't doing it at all because they couldn't figure out a way to execute this in practice without doing it on everybody, which for the same reasons as mine, we're not willing to do. So we asked 2 questions. Can we introduce this standing order in 2 other institutions, and what is the impact on physician behavior and on downstream test ordering behavior? So actually before that, the first question I asked was, and this gets at the principle, how are we actually performing? First question I asked was, are our bone density technologists who have to see if people meet these criteria and execute it at the time the person gets a bone density test, are they actually getting it right? And by and large, for the most part, they did pretty well. I know many will quibble about this and say, gee, they're not doing all that good. Guys, I hate to tell you, I mean, look in our own practices how often we for sure are executing something we say we should be doing. I would put these numbers up against a lot of clinical performance data from others. And then we introduced this at a rural healthcare organization in Wisconsin and at a major academic institution. And again, what we did was a time series study design where we looked at if you had an indication for receiving this image, if there was a good reason to do it, was it actually done? And obviously, in one of these institutions, they had a huge uptake and much slower uptake at the academic institution, who were squawking about all sorts of academic points on this thing. It was a very interesting set of conversations around this, that's for sure. And then we also asked, and again, we asked, are we causing more downstream spine imaging? Why did we do this? Because of the stakeholder of the healthcare delivery system and healthcare payers. So here we're introducing new technology. We're proposing doing a little bit more imaging. And now I see the alarm bells running off in the healthcare peers' heads. Oh my God, this is going to set off a whole cascade of additional unneeded imaging. And so we wanted to address that head on and to look at, well, gee, if the densitometric vertebral fracture assessment was done versus those where we did do it, what impact on subsequent imaging over the next 6 months did we see? And yeah, we did impact it some, but not hugely. And I would argue appropriately. I mean, certainly if you went into the charts of some of these folk and looked at it, gee, I think there's a vertebral fracture, I'm not sure. If we document it, is that actually going to alter what we do for this person? Yeah, or gee, I do see a vertebral fracture. This vertebra looks really funny. There's a lot of density in it. Better rule out a tumor, and hence MRI. So, and then finally we asked, well, okay, now we've done this. Does this actually impact physician prescribing behavior? I mean, if you have a vertebral fracture like this in low bone mass, you're at very high risk of subsequent fractures. And by and large, that's accepted as an indication for fracture prevention therapy. Now, they may be getting fracture prevention therapy for other reasons, hence adjusting for confounders in a logistic regression model, as you see here. But adjusting for all these other indications, an odds ratio of 3.2. So clearly, our readings on these VFAs in all three institutions had an impact in terms of what physician behavior was. So, you know, we did this to really try to really see, gee, do we have good evidence that this is reasonable practice to do? Now I'll show you another example, and this particular example is designed to illustrate the crosstalk between research and quality improvement. And this particular project that I was involved in really involves both of them. This came from one of my partners. Key Pison sends up as a rheumatologist in our division. And he's a very smart guy, very curious, very enthusiastic. And his question was, are patients more likely to experience adverse events if they have gout, if you push allopurinol doses up beyond what is normally recommended for a level of renal function? And the reason why the question came up was that depending on which specialist you asked, you get a different answer. So Key had a patient. This really happened. He had a patient. Comes in, allopurinol doses 100 milligrams a day, gives a person instructions to stepwise increase the dose up to 300. Person goes back to their nephrologist, dose gets lowered to 100. Comes back to Key, gradually raise up to 300, back to the nephrologist, down to 100. Comes back to Key. He gets mad. Comes into my office and says, rah, 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 there's no data to back up what the nephrologist is doing and that kind of thing, and et cetera, et cetera. So we put a project together to try and answer it. So again, this is a question coming from a clinician, wanted to try to get an answer and says, I don't have enough literature support, or I need more literature support to argue with the nephrologist to try to settle this issue between us. And so we decided to do a retrospective medical record review study. We went back 7 years and identified everybody in our institution who had been given an index prescription for allopurinol. And then we had to think very carefully how to define an adverse event. Now this gets at the issue of study design, bias, and threat to the validity of your conclusion. We had to really spell all this out before we did it. We couldn't start the study and say, gee, now we don't like how we defined adverse events. We're going to change the rules of the road midway through. That is just a prescription for getting, biased answers. So we had to really define that a priori very carefully, and then really look at serum creatinine body weight within 6 months of the maximum prescribed dose, so that we could determine, is the dose of allopurinol in excess of what's recommended for this person's level of kidney function or no? You have to have creatinine to know that. Well, we turned up a quality problem. Creatinine was missing in 613. I mean, if you're going to know whether you've prescribed too much allopurinol, you got to know what the renal function is. So anyhow, we then looked at this, and this is just the crude data. And what we saw was that, yeah, we did We did see an increased incidence of definite adverse drug reactions if we pushed the dose. And I should define what these are. Low was if it was within the nephrologist's recommendations for level of kidney function. Moderate was if we exceeded by 1 to 1.5 times. And high was if we went even 1.5 times what the nephrologist told us would be safe to do. So some of these folk, we were really pushing it pretty good. And indeed, there was a slight increased risk, but the absolute risk was still low. And none of these were particularly serious. And just to adjust for confounders, we basically saw really the same thing. And then if we looked at, did we get the serum uric acid below goal consistent with prevention of gouty attacks of arthritis and resolution of tophi, the fact is that even with allopurinol alone, there were a lot of folk where we didn't achieve it. But clearly, if you're dosing down in this region, your odds of success were not very high. So this came out at a time where there was other research from other countries, and that really getting at some of this issue as well. But this is how in our institution we did settle the question between nephrology and rheumatology. I mean, we could have argued anecdote across each other till Timbuktu freezes. They got their anecdotes, we got our anecdotes. And I don't think that so this was simply a way to try to get at a mutual agreeable answer. But, so this was generalizable knowledge, and yeah, we did manage to get this published in a peer-reviewed journal, that you start with a low dose of allopurinol, but you can slowly increase it to get uric acid to goal. But it became a quality improvement project because we turned up quality issues in care of people with gout at our institution. They only had a serum creatinine, and a lot of folk never got to goal. So clearly, we had some major issues here to deal with. And that led to a quality improvement project, again, involving internal medicine and family practice stakeholders. Rheumatology did not just take this on and try to tell everybody else what to do. You know, this project then really got them involved and say, how together do we solve this problem? And it ended up where we had prompts put into the electronic health record. Key put together a document, telling physicians how to dose allopurinol, where you bring up the allopurinol script into the health record. There's a link you can click on it. It gives you the dosing guidelines to try to really help make it. And designed in this way so that in the context of a busy clinical practice day, clinicians will actually execute it and actually do it. So why are clinician scientists necessary? Well, we need better generalizable knowledge for what works best and for whom. There's just a lot of stuff we do out there where we are, to some degree, just flying by the seat of our pants. And the trade-offs of benefits and costs. And we need quality improvement documentation. We're treating patients as we claim we are, and we're achieving reasonable outcomes. I'm sure you're all aware that increasingly payers are demanding this of us. So we're going to have to do it. And clinicians are central to these efforts. In the interest of time, I'm going to get right to these last two, because these are two important slides, I think, to show you. What's in it for those of you who are thinking, gee, I just want to be a doc, I just want to get, I'm going to go into the practice, see my patients one by one, go home, get to my family, just forget about all this. What's in it for you to really maybe think about at least having some really knowledge and skill in this area? Well, at least you have skill sets to critically evaluate evidence that others are claiming to you is true. You get a voice in what problems and uncertainties need to be addressed and what the prioritization of research questions ought to be and how to interpret them. Do these really matter to my patients or no? Do my patients fit these profiles or don't they? Job satisfaction. I mean, there's a lot of stuff out there in terms of physician burnout and in terms of clinicians feeling that they're just really caught in the grind of having to see patients every 15 minutes and so on and so forth. This is a way, at least, to really also try to bring something more to it. Now, what's required? Well, you got to be intellectually curious. I think if you just don't have much intellectual curiosity, I don't think you're going to really find that this is particular for you. Got to be intellectually honest. I mean, for this For this enterprise to work, we have to put ourselves on the line a little bit and design our trials that they really are a fair test of our hypothesis. If you do this long enough, you're going to come across a project where you're disappointed, where your idea that you thought was right is shown to be wrong, or at least likely may be wrong. It can stink a little bit. I've had a recent episode like that where I was convinced was right. It is the most negative study I have ever done. Now, that was a better answer than a maybe answer. A maybe answer where you didn't answer the research question gets at the issue, did you have flaws in your study design to begin with? Sometimes you need your colleagues to keep you honest. I mean, that's partly what we do for each other as well. And just a little pet peeve. Physicians, sometimes surgeons, sorry if I'm picking on surgeons. I've had some come to me and say, gee, I'd love to do this, but it just doesn't pay and that kind of thing. And I go, I'm sorry. But no, research budgets can't pay you what you earn in OR. Not going to happen. Instead, I would encourage all of us to think, what do we take home at the end of the day from our jobs? What's important to us? What makes us feel like we made a contribution? And I think that successful clinician scientists are going to be folk where they're taking something home a lot more than just their paycheck. And I'll stop there, and I'm sorry I ran over, but I'm happy to take any questions. Yes?
So I want to know what you would do as a clinical scientist. So this is an hypothetical scenario where you conduct a clinical experiment with a terminal cancer patient. So you have a new drug. So you want to put the terminal ill patient in placebo group and experimental group. Now setting aside all the ethical issues, so you continue with the experiment. But after a while, you see the experimental group actually improve, but on the other hand, the placebo group is deteriorating. So in that condition, what you would do, whether you switch the patient from the placebo group to the experimental group, because there are here other things. But if you do that, that will affect your quality improvement, skew your data, so you want to stick to the scientific part of it. Or you say, oh, you don't qualify anymore, so you are on your own, so please leave. Or 3, being a doctor, whether it is you have a moral obligation to save that patient, so it doesn't matter, even if it affects my science, I have to switch the patient from the placebo group to the experimental group because I want to save the patient. So what would you do if you are a clinical scientist?
Yeah, it's a great question. And I would answer it at 2 levels. I'd answer it for the study research team and funders from that perspective, and from that patient and their individual physician caring for them. So from the research team's perspective, this is why big studies have data safety and monitoring boards. And one of the job of the data safety and monitoring board, they get data like every 6 months or so saying, not only is the study on track recruiting people, but are we seeing unfortunate safety signals in either treatment group, or are we in fact seeing that in fact there's an endpoint? I don't remember it now off the top of my head, I'm sorry. There is a recent example out here where a study was terminated by the funder, by NIH, early, because exactly what happened was what you stated. They said, look, we don't need to go beyond this. We have enough here where we're confident enough, even from the scientific perspective, that we've answered the question, that we're going to stop. Now, if they haven't got enough data to really have complete confidence that they've got a firm answer, now you're in a grayer zone. Well, am I putting my patient more risk to keep them in the control group? Well, maybe, maybe not. We don't have enough yet where we're on firm, complete scientific grounds and evidence to say for sure we know what the highly likely answer is at a very high level of probability. However, then there's the individual patient and their physician perspective. And there is nothing wrong from my perspective from that physician saying, look, the trial's going to go on because we're not sure. But my sense is that it is in your best interest to withdraw from this study and go on to do something different. And I can tell you that one of the things as IRB chair we look for in every consent is that it is stated that patients can do that without any penalty. So I don't know if that fully answers what your question was. Usually they're not going to, the study sponsors of that are not going to say for sure we're going to go ahead and continue and that kind of thing. I've seen a couple oncology trials where it was really for safety reasons, where the Data Safety Monitoring Board was picking up, oh gosh, it came before my committee a month or so ago, and actually it was excess mortality in the experimental group, and they just stopped it. Trial was done. But that's why you have to have that kind of monitoring on those studies.
I had a question concerning your quality improvement studies. How do a lot of these requires behavior change. In other words, someone's going to have to do something in their practice. So how do you ensure high fidelity with regard to the behavior change that you might want in a particular study?
Yeah, another great question and a very difficult one. And the science of behavior change itself is evolving. So we don't necessarily have great answers to that. But no, it is a critical one, and it's one that any really well-designed practice intervention study, I think, has to look at. That's why we felt in our vertebral fracture one, it was really important that we really look at, gee, are we having any impact on at least physician behavior? Now, that's still a limitation in that study is still that I don't know what effect that had on patient behavior. I don't know what effect that had on outcomes. I can't tell you that we've impacted the secondary fracture rates, for example, in these people by doing it. That would require a different investigation to do it.
Perhaps this would require another hour presentation, but I was wondering if you would comment about how this big overview that you've so elegantly sketched out for us would apply to the challenges we face in osteopathic manipulative medicine research. For example, I was struck by the SPRINT study that you cited, and we've seemed to have butted our heads up against the wall for decades with regard to issues of placebo control and so forth. And in the SPRINT study, you explicitly stated that neither the patients nor the practitioners were blinded. And whether it was that, and I think it was that study as well, where it wasn't really a matter of comparing a specific drug to a placebo, but a whole intervention, and a whole which is comparatively amorphous compared to a specific drug. So I wonder if you have any brief comments about that.
Yeah, no, I would think in terms of strategies, of treatment strategies where a variety of techniques are used, and where certain manual medicine approaches are used for certain patients, and you decide instead I'm going to use a little different technique in this particular thing. Again, I think that there are going to be trade-offs. So, when you're doing multiple things, you won't be able to say, well, gee, it's because we did X additional amounts of this particular type of manual treatment versus that, and when you have kind of a strategy approach like this. So it's going to be really, really hard to tease that out. But, I think this is applicable for the kind of settings that you were talking about. And in terms of when you think of more broadly, in fact, impacts on patient behavior, it is going to be very often these sorts of treatment strategy approaches. I mean, one of our researchers has a very large grant from the Patient-Centered Outcomes Research Institute looking at pharmacist management of blood pressure. Now, here's an example where here the intervention is simply a practitioner seeing a person additional, not driving specific medications, and this kind of thing. So there are certain questions that's going to answer and certain ones that aren't. It just requires critical thinking as you design it. What am I going to be answered with this study design, and what am I not going to be able to answer with this study design, and which are the questions that really take priority?
Okay, thank you very much. Certainly, there'll be opportunities during lunch to have further questions answered. Everybody is invited to go upstairs for our free lunch before we start our poster session. So thank you all very much, and we'll see you upstairs.